The program. So welcome all of you to this truly special event in honor of our friend and colleague, Chris Walsh. I'm delighted to have all of you here, and I would like to just start out with a few thank yous. So I want to acknowledge the work of the organizing committee for this teaching event. Wenjun Zhang, Michael Marletta, Tom Wanless, Jason Sello, Denitza Fujimori, with lots of support as well from Bria Castellano and Danielle Vitale from ChemH for putting together this program today. I also want to say a very special thank you to our speakers. This has been a really fun symposium to put together because this is not a typical type of research event where you come up and you present something from your lab or some project that you've worked on directly. It's a teach-in. And so it's been all of the talks you're going to see today are brand new. We've had those who work directly with Chris paired with trainees who perhaps never met Chris, working together to cover different topics that Chris really shared some beautiful light on during the time of his academic career. So I just want a few words to begin. I think Chris, like so many of us, wore a large number of hats. He was a friend, a colleague, a family member. But I think what's really unites us all here together today, some of us knew Chris directly, worked with him, worked in his lab, collaborated with him. Many of us did not. Many of you, I'm sure, never met Chris. But still, the thing that we all have in common is that we are all one of Chris's students. One piece of advice Chris always gave me over and over, I heard many times, was that if you really want to learn something, you need to teach it. He did this time and time again, not only in his lectures or in the classroom, but in his writings and his textbooks. And I think this really reflects something that was a motto for Chris, that almost counterintuitively, being a teacher is really synonymous also with being a student. As a lifelong learner with endless depth of curiosity, I think Chris continues to teach us even today. So please sit back, relax, and enjoy Chris's living legacy in the talks that we'll hear throughout the day. Thank you. We have multiple sessions. I'm going to ask Chetan Khosla to come up and get us started with our first round of talks.
So thank you, Beth, and thank you for all the hard work that you have done together with Michael to make this happen. I want to add a somewhat distinct but complimentary welcome to all of you who are Chris's students, who have come over here, whether you trained with him as doctoral students or postdoctoral trainees or even undergraduates. I'm told that there are 200 people live streaming this event too, and so I suspect there are a number of you who consider yourselves as Chris's students. So this event is for you. As Michael said when we decided to put this event together, it's all about beginnings and endings, as Chris told him. We're going to make this event a really special one. And as Beth pointed out, there have been several people, preceptors and trainees who have worked incredibly hard over the past several months. And so without further ado, I'd like to start by introducing the first pair. The first pair is Emily Balskus from Harvard University, who was with Chris, about 10 years ago as a postdoctoral fellow, and Catherine Liu, who is currently a postdoctoral fellow here at Stanford. who finished her PhD at Stanford as well not too long ago. They will be talking about a subject that, as all of you remember, is essentially the sine qua non of the Walsh legacy, scientific legacy, the chemical logic of biology. So without further ado, I'll let Catherine and Emily take over. Welcome.
Okay. Thank you so much, Chetan. And again, thank you to everyone involved in the organization of this event. It's wonderful to be here and have this opportunity to remember and celebrate Chris's scientific legacy. So it's my responsibility to begin the program by discussing Chris's contributions to elucidating the chemical logic of biology. So this is obviously a big topic. So drawing from Chris's own perspective and writing, I'm going to try to convey the impacts of this way of thinking, both on the field of chemical biology and also on myself personally. And in doing so, I hope to provide a broad introduction to Chris's scholarship that will set the stage for the presentations to come. So Chris himself highlighted that a desire to understand the chemical logic of biology was the central question that drove his research. And specifically, he was intrigued by metabolism, the set of chemical transformations that are carried out by living organisms. But what do we actually mean by chemical logic, and why is this perspective so powerful? Well, here I show a image of a map depicting metabolism in cells. Chris described these types of maps as posing stultifying complexity. And I think that quote also really nicely describes how I thought about biology and metabolism as a graduate student studying organic chemistry. Metabolism was hopelessly complicated, just a set of names and pathways that one simply had to memorize. But this all changed when I joined Chris's lab. Chris taught me and the field the power of applying chemical knowledge and principles to understanding biological systems. And so to really illustrate the concept of chemical logic, I'd like us to consider three small molecules that play central roles in biology: ATP, NADH, and acetyl-CoA. So on the surface, just looking at these molecules, it may be hard to understand why they're so important in biology. But if we begin to think deeply about their chemical properties, about their structures, their reactivity, we start to recognize that there's a chemical basis for their utility in living systems. We start to understand why they participate in certain types of chemical reactions, and we start to recognize these patterns of reactivity being used over and over again throughout metabolism. We can also start to contemplate the chemical features that these three molecules share in common. And so, as Chris has highlighted in his writing, these molecules share an important chemical feature. They are all reactive enough to enable important reactions in cells, but at the same time, they're stable enough to persist for periods of time in a cellular setting. And it's this critical chemical property that perhaps explains their ubiquitous roles in biology and underlies their use in thousands of biochemical reactions. So to me, chemical logic means considering biological phenomenon from the perspective of a chemist. And this way of thinking can be applied to many aspects of biology. And one particularly interesting problem is the problem of understanding enzymes, the protein-based catalysts that perform chemistry in cells. Of course, Chris was intrigued by enzymes, beginning with his graduate research, and I actually wanted to share some of his work from this time. And so, as a graduate student, Chris worked on the enzyme ATP citrate lyase, which performed what was, at the time, considered to be a very complicated chemical reaction. It takes a 6-carbon molecule citrate and breaks it apart into a 2-carbon product and a 4-carbon product. So this is a carbon-carbon bond cleavage reaction. And so it was known at the time that this reaction involved a thioester intermediate. And this made good chemical sense because the thioester could serve as an electron sink to facilitate breakage of that carbon-carbon bond. But the question that intrigued Chris was how one could form this thioester intermediate from citrate, which does not have an electrophilic functional group. And so he hypothesized that the role of ATP in this reaction would be to form an intermediate called citral phosphate, as this intermediate would have the chemical reactivity needed for thioester formation. And so by thinking deeply with this chemical perspective, Chris was not only able to generate this hypothesis, but design experiments to test it. So, of course, Chris would continue to apply this type of deep chemical thinking to the study of many other enzyme mechanisms over the years. And beginning with his postdoctoral work and early academic career, he became very intrigued by groups of enzymes that work together with small molecules. And so these small molecules are known as coenzymes or cofactors, and they expand the reactivity of enzymes by giving them reactivity beyond what is available to the protein scaffold itself. So Chris found these cofactor-dependent enzymes to be exciting starting places for all sorts of discoveries. For example, in characterizing the flavin-dependent enzyme, cyclohexanone monooxygenase, he learned that the reactivity of cofactor-dependent enzymes could mirror reactions that up until that point had only been developed by synthetic chemists. In also studying flavin-dependent enzymes and pyridoxal phosphate or PLP-dependent enzymes, like alanine racemase, Chris recognized that an understanding of enzyme chemistry could help guide the design and study of synthetic molecules like trifluoroalanine that can interfere with enzyme chemistry, so-called mechanism-based enzyme inhibitors. Of course, the synthesis of small molecules that inhibit enzymes continues to be a huge focus of modern chemical biology and drug discovery. But Chris also recognized there were opportunities to apply chemical thinking, not just to synthetic molecules, but also naturally occurring bioactive small molecules called natural products. So it's the structural features of natural products that give rise to their fascinating and varied biological activities. And this structure-function relationship, I think, was a driving force behind a lot of Chris's work on natural product biosynthesis. One beautiful example is the biosynthesis of the iron-binding natural product enterobactin. And so it turns out that it's the catechol building blocks of this natural product that give it the ability to bind iron tightly, sequestering it and fueling bacterial growth under iron-limiting conditions. Likewise, a chemical understanding of the antibiotic natural product vancomycin, and specifically an understanding of how this molecule interacted with its target in the bacterial cell wall, allowed Chris to recognize how this antibiotic loses its efficacy when drug-resistant bacteria are able to change the structure of their cell wall. So Chris also recognized that many important chemical reactions in biology don't just involve small molecules. They can involve much larger molecules. And so he became interested also in the study of post-translational modifications. And so these are chemical transformations that occur on proteins and peptides that alter their structures and their biological functions. So one very prominent example of a post-translational modification Chris studied involved the addition of coenzyme A to carrier proteins. And so this installed a conformationally flexible group onto the protein scaffold that also contains a very good nucleophile. This makes the carrier proteins, after they're modified, perfect tethers for substrates, intermediates, and products on enzymatic assembly lines. Chris also showed us how post-translational modifications can completely morph the structures of peptides, generating, for example, complex, rigid, heterocyclic scaffolds like thiocillin that also possess potent biological activity. And I think this second example highlights that some of the most amazing opportunities to apply chemical thinking within biology lie in studying how multiple enzymes work together in the context of biosynthetic pathways that build natural products. So an interest in the chemical logic of biosynthesis was a main focus within Chris's lab when I joined. And perhaps most notably, his work revealed how enzymatic assembly lines, like polyketide synthases, and especially non-ribosomal peptide synthetases, used the coordinated action of many enzymatic domains to build complex molecules. So Chris described these pathways as unrivaled by and inspiring the most creative cadre of synthetic chemists. And indeed, I think it was this work that actually drew a lot of synthetic chemists, including myself, to want to join Chris's lab. But Chris also realized that the idea of chemical logic didn't just apply narrowly to individual biosynthetic pathways. One could also uncover chemical strategies that transcended multiple classes of natural products and enzymes. And a beautiful example of this is cryptic halogenation. So Chris had this amazing ability to spot these types of common themes in natural product biosynthesis, and also for recognizing when biosynthetic strategies paralleled strategies that were used in chemical synthesis. So clearly, Chris's work shows us how seeking to understand chemical logic can inform discoveries in many areas of biology. But I believe that the impact of this way of thinking extends far beyond these individual discoveries. Chris, of course, brought this perspective into the classroom, recognizing that exploring the chemistry of living organisms was a powerful way to engage young scholars. And so when I was a postdoc, I had the opportunity to help Chris with a class he was teaching to medical students. And I am still so impressed by his clarity of thought and the incredible organization of his lecture notes. He also left a lasting imprint on teaching in my department at Harvard when he developed the curriculum for Chem 27, the organic chemistry of life. And this is a course we still teach that introduces the chemical logic of enzymes and metabolism to undergraduates. And it was this desire to educate, this interest in teaching that first inspired Chris to capture and share his chemical perspective with a broader audience in the form of the many books and articles that he authored. And this, of course, further extended his impact as an educator. But I also want to emphasize that this focus on chemical logic also guided Chris's own learning together with members of his lab. And this is because chemical thinking provides a powerful framework for problem solving and hypothesis generation. So time and time again, when we would be discussing a puzzling result or a seemingly insurmountable research obstacle, we'd meet with Chris and he'd urge us to focus on chemistry. He'd pull out the folder where he kept individual notes on our projects or one of his index cards, and we'd write out mechanisms. We'd draw structures. We'd contemplate reactivity. And in this way, we could more clearly define the problem. We'd recognize parallels we hadn't seen. We'd come up with ideas for new experiments. And so this process provided me and so many others with an invaluable education in both science and mentorship. And I want to also highlight that a very important lesson I learned from Chris during these types of conversations is that a chemical perspective doesn't just help us understand what's already known in biology, it also helps us explore the unknown. For example, we can use an understanding of the chemical logic of metabolism to recognize when natural products contain structural features that can't be explained using known enzymatic chemistry. We can then use chemical thinking to develop biosynthetic hypotheses that guide searches for the genes encoding the enzymes that build these molecules, fueling enzyme discovery. We can also use chemical thinking to go in the opposite direction, beginning from uncharacterized gene clusters, predicting the types of reactions they might encode, and in that way guiding the discovery of new natural products. So right now, this powerful combination of chemical logic and genomics is allowing the field to explore nature's biosynthetic capabilities at unprecedented speeds and scale. And it's really Chris's contribution, I think, that have enabled this exciting new era in natural products research. But I think this way of thinking is also critical for the field of biology as a whole. And so Chris described the evolution of modern biology as beginning with the discovery of small molecules from living systems, progressing to a focus on understanding macromolecules like DNA and proteins, before finally reaching the current multi-omics era of biology, where we have access to a wealth of information about living organisms in the form of genomes, proteomes, metabolomes. But now the critical challenge that we face is how do we actually make sense of all of this information? So I think applying chemical logic is going to be critical for solving this problem, not just for allowing us to learn about biological functions, but importantly, to help us target those functions therapeutically. And so I think that Chris's work, his way of approaching research is going to continue to drive discoveries at this exciting interface of chemistry, biology, and medicine for years to come. And so now I'd like to end by sharing one final impact of Chris's contributions to elucidating the chemical logic of metabolism. So I think we've all been transformed at various times in our lives by experiences and perspectives that alter our view of the world. So for many of us, we might think of experiencing art, architecture, or music. And here is a piece of art that I hope Chris would have found inspiring. So I believe that through his research, his teaching, and his mentorship, Chris changed our perspective on the world. He showed us how to view the world from a chemical perspective, allowing us to appreciate it in a completely different way. So I'd like to thank Chris for sharing his vision, for sharing his passion for discovery, and allowing us to accompany him on his journey to reveal the beautiful chemistry at the core of nature. And with that, I'd like to invite Catherine Liu up to the podium to share some of Chris's research.
Thank you so much, Emily. That was an incredible perspective. So I guess, sorry, we're going back here. But hi, everyone. My name is Catherine. I'm in the Satley lab, and I'm so excited to be here with you today to share a story that I think perfectly highlights some of the points that Emily made about how understanding the chemical logic that underlies biology can really help enable discovery. So I'm going to share with you a paper today that came out of Chris's lab when Emily was there, and it's about the genetic and molecular basis for sunscreen biosynthesis in Cyanobacteria. So to start off with a little bit of context for those of you, myself included, who don't spend a lot of time thinking about cyanobacteria. These are actually really amazing organisms, and that's because they were one of the first life forms present on the Earth. And even though they're tiny, they actually have enormous ecological importance. And that's because they were one of the first organisms to learn how to do photosynthesis. So they're able to take energy from the sun and use that to fix carbon and produce oxygen. That led to oxygenation of the atmosphere, and long story short, all life as we know it today. But there is a downside to depending so heavily on the sun as an energy source. And just like when we spend too much time in the sun, our skin can turn red and we can get skin cancer. Cyanobacteria can also experience lethal DNA damage as a result of UV exposure. Now, pretty amazingly, these bacteria have adapted and developed strategies to protect themselves from the sun's UV. And one of these is making small molecules that act as sunscreens. One type of these sunscreens are called mycosporines and mycosporine-like amino acids. I've shown a couple of these structures up here. And these molecules are really amazing sunscreens for a number of reasons. First of all, they happen to absorb light in just the right range to protect from UVA and UVB damage. Second of all, they don't tend to degrade after exposure to UV light. And finally, they're able to safely dissipate the energy that they absorb without producing harmful side products like reactive oxygen species. In fact, these molecules are such great sunscreens that they actually have been included in a number of commercial formulations for human use so that we can pretend to be cyanobacteria and protect ourselves with their molecules. So the secret to why these molecules have such great properties actually lies in their chemical structure, as Emily alluded to. These mycosporines and MAAs both contain these vinylogous amide groups that I've highlighted in red. And these functional groups directly allow the molecule to absorb UV and also dissipate that energy. So even though these chemical features directly enable the function of these molecules, it wasn't clear at the time that Emily and Chris started this project where these molecules even came from. And when we think about the types of molecular building blocks that are available to cyanobacteria, they're usually molecules that are already floating around for primary metabolism that the bacteria need for everyday function. So I'm showing things like amino acids, things that amino acids come from, and also products of carbon metabolism. Now, when you think about it, it's pretty amazing that these bacteria are able to assemble these very fundamental building blocks that many other organisms have access to and build them into such highly specialized, highly effective molecules that directly enable their own survival and fitness. And so it was exactly this question that Emily and Chris wanted to tackle, which was how are cyanobacteria making these MAAs? But starting off with a very unfamiliar looking molecule, with the cynorine shown here, and working in a pretty complex bacterial system, how do we even begin to understand the types of enzymatic steps that can bring us to this molecule? So Emily and Chris started by breaking this molecule down into more recognizable chunks. They saw that cynorine has a couple of amino acids attached to it. And there are reactions in the cell that are known to attach amino acids onto molecules. And so they thought that whatever set of enzymes is responsible for making cynorine probably includes a couple of these amino acid attaching enzymes. When we take off the amino acids from cynorine, we end up with a molecule called deoxygadosol, which I'm going to call DG. Emily and Chris further noticed that DG itself contains a very recognizable functional group, an O-methyl group, and so they added an O-methyltransferase to the list of enzymes that they thought might be involved. And taking that methyl group off of DG gives a molecule that I'm going to call desmethyl-DG. And Emily and Chris noted that desmethyl-DG looks a lot like another molecule that they knew from primary metabolism. This is DHQ from the shikimate pathway and is involved in making aromatic amino acids. And so by understanding the types of reactions that a cell can do and the logic behind assembling those steps together in a metabolic pathway, Emily and Chris were able to assemble a short list of enzymes that they thought were involved in cynorine biosynthesis. And when they looked in the genome of Anabaena variabilis, which is a cyanobacteria that's able to make cynorine, they were able to find a gene cluster that encoded for those exact expected activities. So this gene cluster included two enzymes that were associated with amino acid attachment, including an NRPS-like enzyme and an ATP grasp. The gene cluster also encoded for an O-methyltransferase. And finally, there was also an enzyme that was homologous to another enzyme in primary metabolism in the shikimate pathway. And when they expressed this entire gene cluster in a heterologous host, E. coli, they found that the E. coli was suddenly able to make cynorine. So it turns out this gene cluster had all the machinery needed to make cynorine and MAAs in a bacterial cell. Now, Emily and Chris very carefully and painstakingly characterized each of these steps in the pathway but in the interest of time, I'm going to focus on two that I think really highlight how understanding chemical biology and chemical logic can help us understand new biology. So I'm going to actually start at the beginning of the pathway with this enzyme AVA3858, which was noted to be a homolog to an enzyme in the shikimate pathway. So Emily and Chris knew that this enzyme was making the molecule des-methyl-DG, but it wasn't clear what the substrate to this reaction was. But at the same time, they did note that this enzyme in its product looked very similar to other enzyme in product pairs from bacterial metabolism. One of them, I've already mentioned before, is DHQS from the shikimate pathway. But another homolog is EVS, which takes intermediates from another pathway in primary metabolism, the pentose phosphate pathway, and turns those compounds into molecules that look a lot like des-methyl-DG. And so to find out what substrate AVA3858 used, Emily and Chris decided to test their new enzyme with the substrates of its homologs. And what they found was quite amazing. It turns out AVA3858 doesn't seem to use intermediates from the shikimate pathway, but instead uses the exact same substrates as the homolog EVS. And you'll note that even though AVA3858 and EVS perform very similar chemical reactions, and they also share the exact same substrate, they don't seem to make exactly the same product. On top of what EVS already does, AVA3858 further eliminates the hydroxyl group that I've highlighted in red and also forms an additional double bond. And this double bond is a really important step in making the cyclohexene ring that ends up helping UV absorbance in the final molecule. So to summarize this step really briefly, Emily and Chris were able to recognize the shared patterns behind the reactivities of these enzymes. And they used that approach to find out that Schnorren biosynthesis starts with pentose phosphate pathway precursors. And not only that, but I think this is a really awesome example of how an enzyme can closely resemble another in primary metabolism, but because of a slight added functionality, it suddenly feeds a whole specialized branch of metabolism. Okay, the second step that I like to focus on today concerns attaching this glycine amino acid to the DG core. And this step is interesting for a number of reasons. First of all, this step forms an enamine moiety, which I've highlighted in red here. And this enamine is really important for enabling UV absorption in the final molecule. In fact, changing out the glycine at this position for other amino acids can shift the wavelengths at which the final molecule can absorb. So it's functionally important to understand the biochemistry of this reaction. Second of all, enamines and imines are known to be made by very specific enzyme families. These are usually ATP-independent enzymes, including things like class I aldylases and PLP-dependent enzymes. And what Emily and Chris found striking here was that it wasn't those usual suspect enzymes performing this reaction, but instead an ATP grasp enzyme. ATP grafts are actually a pretty large enzyme family, and they're known to form a bond between a carboxylic acid and another molecule. And just to show you a more concrete example of what that looks like, I'm going to use the very classic ATP grasp example of a dealanine ligase. This enzyme connects two alanine molecules together with a peptide bond. So how this enzyme works is that it first activates the carboxylic acid of one of those alanine molecules by attaching a phosphate group to it. And this phosphate group is donated by an ATP molecule. A nucleophile, in this case, the amine group of another alanine, can then come in and form that peptide bond at that highlighted position. Now, at first glance, it might seem a little surprising that mycosporine glycine is made by an ATP grasp. After all, the carbon-nitrogen bond that's formed here looks nothing like a peptide bond. But once Emily and Chris looked more closely at how these ATP grasp enzymes worked, they saw that the parts needed for the enzyme reaction to occur, like the carbonyl group and the hydroxyl group, are all there on the DG substrate. They're just separated by a little more distance on the molecule. And so when they performed further mechanistic studies, they saw that the ATP grasps in the mycosporine pathway act completely with the same logic as a canonical ATP grasp. So in this enzyme, This enzyme first starts by activating the hydroxyl group with a phosphate group donated by an ATP. A nucleophile, in this case the amine from the glycine, can then come in and form the bond that I've highlighted there. So Emily and Chris were not only able to show us more about how MAAs are made, but also expand our understanding about the types of reactions that ATP grasp enzymes can do. It was previously thought that these enzymes mostly act on carboxylic acids, but here Emily and Chris showed us that they can also work on vinylogous acids. And this opens up a whole new world of different substrates and functionalities for this large and important enzyme group. So just to summarize here what we've learned today from Emily and Chris's fantastic work, they were able to uncover how cyanobacteria make a very chemically interesting, but also ecologically important and commercially important cyanobacterial sunscreen. Along the way, they ran into a few enzymes that didn't seem to work in expected or predictable ways, but they all seemed to follow a fundamental chemical logic. And Emily and Chris used their understanding of this chemical logic to expand our own understanding of what these enzymes can do and also their families as well. So thank you so much, and thank you also to Emily for letting me talk about her wonderful work.
Okay, we'll do the following. We have coffee, and I know that that's something that many of you desperately need. So we will start by, if anybody has a specific question for Catherine about the paper that she presented so beautifully, now would be a good time to ask. And then we'll take a coffee break, and then you can come back and continue the discussion for a few more minutes with Emily. How does that sound? Okay, well, Catherine's here to educate you in the true spirit of Chris.
Any questions?
Carolyn.
That was a beautiful presentation. Oh, thank you. That was a beautiful presentation of Emily's postdoctoral work. That's really awesome. And a question that comes to mind is, since that study was published, which was 2010, I believe, it's been 13 years, and those enzymes that were uncovered back then, ATP grasp acting on a vanilligus acid instead of just a carboxylic acid. I mean, now have related enzymes to those come up where you can make even another layer of predictions based on that knowledge?
That's a great question, Carolyn. From what I understand from the reading, since the paper came out, there have been a number of other works that have published about mycosporine biosynthesis in cyanobacteria. Turns out, it appears there are multiple pathways that these bacteria actually use to make these compounds. So this is one of them, but there are multiple strategies. Regarding specifically ATP grafts in those substrates, I'm not entirely sure about that, but I wonder, sorry, Emily, if you had any perspective on this.
Yeah, sure, there are definitely, okay, yeah, sorry.
Oh, you have a mic on. You'll be
Yeah,
okay.
I have a mic on. Yes, there are definitely variations on this biosynthetic theme that still involve ATP grasp enzymes. So there'll be some pathways, I believe, that use 2 ATP grasps as opposed to just one to make both carbon-nitrogen bonds. But what's been very exciting since that time, I think, in the area of ATP grasp chemistry is just that we can find so many more examples within biosynthetic pathways that do interesting variations on chemistry that involves ATP. So not just kind of making peptide-like linkages, but making other types of linkages as well.
Cool. Great question. Thank you. Michael.
So what do we know about maybe the photoactivation chemistry of these molecules? How do they protect the cyanobacteria?
That's a great question. I believe, so I think it's actually still a pretty active area of investigation. I think the last paper I read about the mechanism of photodissipation was in the 2020s. So I think it's still a very active area. So, but I think they have identified through structure-activity relationships that the groups that I pointed out, like the vinylogous amides, are really key chemical features that are associated with that.
Let's take a coffee break and then we can come back. And if anybody has questions or thoughts to share with Emily, I'm sure she'd be happy to continue the conversation. Yeah, good morning. Can I ask everybody to get back into your seats or however you wish to spend the next hour? And I'd also like to invite Emily up here in case there are any burning questions for Emily. Does anybody wish to have any discussion with Emily before we move up to the next session? Clearly, that paper was a great kickoff for the day. So, any questions, thoughts, comments to share with Emily? People are too busy with their coffee. Laura.
So, really beautiful work, Emily. Are those gene clusters found outside of cyanobacteria? And could other microorganisms be making similar molecules?
Yeah, so that is a wonderful question. And these naturally occurring sunscreens are actually really widely distributed, not even just among bacteria, but among life in general. And so, since that initial gene cluster discovery paper, there have been gene clusters discovered in a variety of different organisms that were known to kind of have molecules like these. There's some interesting examples of marine eukaryotes like sea anemones and even zebrafish who have part of the pathway and can make some of the earlier intermediates that are thought to be antioxidants. With organisms like sea anemones and corals, it's thought that there could potentially be collaboration with microbial companions to construct the actual sunscreens. But yeah, these molecules are ubiquitous across marine life, and that's one of the reasons why they might make great sunscreens, because they're obviously present already in marine habitats at really high levels.
David has a question.
How much do they need to protect themselves? The cyanobacteria, is it like of their dry weight 1% or a small percentage? Are they really, really potent?
Yeah, so for broader context, cyanobacteria make a couple of different types of sunscreens. These are ones that accumulate in the bacterial cytoplasm. And I believe in some cases they do reach percentages of dry weights of 1%, several percents. They accumulate at high concentrations. Cyanobacteria also make usually a couple of different MAAs. So there'll be different amino acids and nitrogen-containing parts of the molecules that will be appended to different MAAs. And that changes the lambda max. And so by accumulating multiple versions of MAAs, the cyanobacteria kind of achieve broad spectrum UV protection.
Okay, well, thank you all, and thank you, Emily, for getting us off to such a wonderful start. Our next session takes us to Professor Walsh, the enzymologist. And clearly, if those two words don't fit right next to each other in your cerebral cortex, then you're in the wrong room this morning. Those two words are basically, well, they're right here on the book. So don't take my word for it, look at the book. Our preceptor-trainee pair is comprised of Professor Sarah O'Connor, who understandably can't be here in person, but is here in spirit and will be here digitally in a moment. And The trainee is Dr. Maria Maya Garzon, a postdoctoral fellow in our colleague John Long's lab and an accomplished chemical biologist in her own right. So may I ask that Sarah's video be turned on, please?
Hello, everyone. I'm so happy to be here, even if only virtually. And I would like to talk a little bit about what Chris Walsh taught us, more specifically me, about mechanistic enzymology, and also how mechanistic enzymology connects to natural product biosynthesis. So when Beth gave me this assignment, my first impulse was to go and to try to find Chris Walsh's first paper out of his independent laboratory. And I think I was hoping to understand from reading this paper how Chris's extraordinary career began. So as far as I can tell, this is Chris's first paper out of his independent lab at MIT. And in this paper, he describes the purification of a flavoenzyme, hydroxy acid oxidase, from rat kidney. And when I first read this abstract, I thought, well, maybe this isn't the best paper to talk about for mechanistic enzymology. But this is Chris, so of course there's a really interesting twist to it. And in this paper, he shows that actually some flavoenzymes only have half of a flavin cofactor per substrate, which was a really cool and interesting discovery. So in this paper, the message that I really took away from it is the first rule of mechanistic enzymology that I learned from Chris, and that is to pay attention to detail. And I think this is best exemplified in this paragraph of the paper here, where they're talking about the details of the purification. And they say that during the later stages of the purifications and of all preparations, the visible yellow color of the hydroxy acid oxidase, the flavoenzyme, seemed low compared to other flavoenzymes that we have previously isolated. So what this means is Chris could look at a solution of a partially purified flavoenzyme and say, there's actually not as much flavin in that as I've seen previously. Maybe we should investigate this more. I would have loved to have been in the room when Chris looked at the solution of that flavoprotein. But absolutely amazing. I would not be able to look at a solution of flavoenzyme and be able to tell you the stoichiometry of the cofactor. Now, just for the students and the postdocs in the audience, this is what purifying proteins from rat kidney in 1975 looked like. It was all native purification, a series of columns and ammonium sulfate precipitations and so forth. So a lot of rat kidney, a lot of time in the cold room to get this beautifully purified protein from this not so easy to work with source. And once this purified protein was in hand, Chris and his coworker did what I thought was an extremely simple but very incisive and definitive mechanistic experiment to measure the stoichiometry of the flavin in this flavoprotein that they had isolated. What they did was they used a suicide substrate called hydroxybutanoate, and that is described elsewhere, as they say in this paragraph. And Lola will actually go into detail in this in the next talk. She has a beautiful presentation on how this suicide substrate works. And this suicide substrate inactivates, it covalently modifies the flavin of the flavoprotein. So they could make a radio-labeled version of this 2-hydroxybutanoate substrate or suicide substrate, and then they could allow this to react with and completely inactivate their isolated and purified enzyme, and then they could purify it on a Cefidex gel filtration column. And then they could measure the milligrams of protein that they recovered and correlate that with the DPM, the radioactivity of the modified flavin. And then using those two measurements, they could definitively show that a ratio of 1 to 2 flavin to subunit was obtained. So indeed, this protein did have half the amount of flavin as would have been expected compared to all other flavoproteins that had been previously worked with. So Chris's initial eyeing of the solution of this flavoprotein exactly matched these beautiful experimental mechanistic results. And this is some of the data here. And again, you'll hear more about this in the next talk. So this was a first paper, very simple. It was a protein purification, but it had a really interesting twist to it thanks to Chris's keen eye and beautiful mechanistic insights. And this paper was most recently cited in 2019. So it's still impacting science today. Now, fast forward 25 years, and this is where I came on the scene. So this is where, I had direct experience with Chris's mechanistic insights. And 25 years passed. This was about 2000, 2001. And this hot new anti-cancer agent had come on the scene, this molecule called apothalone. And everybody was interested in synthesizing it, and making it, and using it, and getting it ready for clinical trials. And Chris had teamed up with Chetan Khosla and David Kane, and they wanted to have a consortium to uncover and elucidate the biosynthesis of this really interesting molecule. And Chris was in particular really interested in the thiazole starter unit, which was created by 3 modular proteins, which were a mix of non-ribosomal peptide synthetases and polyketide synthases. And you'll hear more about this assembly line logic later in the day. So I was a postdoc interviewing in Chris's lab, and he asked me what I wanted to work on. And, I pretty much said, whatever you tell me to. And he suggested that I work on apothalone. I said, great. And I remember I was sitting in his office, and he was drawing out his predicted mechanism for how this starter unit of apothalone formed in his office. And he had these pads of graph paper, and he just drew out this beautiful mechanism and then just gave it to me. And I no longer have those sheets of paper. I was trying to sort of, remember this a little bit in my office the other day. I still try to write things on graph paper in an attempt to emulate Chris. So the graph paper is a little bit, it's still a little bit important to me. Now, in typical Chris fashion, you can see the first paper on apothalone biosynthesis was published in September of 2001. So this was less than a year after they started working on apothalone. So typical Chris. And Raymond Chen, the first author of this paper, along with myself, we managed to crack the first part of the biosynthesis of this thiazole starting unit. And it was exactly as Chris had predicted in his office. You can see this beautiful formation of this thiazolene ring, and then this flavin cofactor, which he had beautifully introduced me to when I was interviewing in his postdoc office, oxidizes the thiazolene ring to the thiazole via this flavin-containing oxidase domain of the non-ribosomal peptide synthetase module protein. And Raymond was able to purify this protein, only now we had histidine tags, so it was much easier than what he had to do with rat kidney, and could show using spectroscopy that we had the typical absorbance that indicated that a flavin cofactor was present. There were also, I just want to highlight other lessons beyond the flavin cofactor chemistry that I learned in the course of working with apothalone biosynthesis. And my job in this project was to work with the starting module, EPO-A, which is a polyketide synthase, and that just loaded the acetate group onto the thiazole starting unit. And I could never get this protein to express. I kind of banged my head against the wall for a while trying to get active protein being heterologously expressed. But what I was able to do was I was able to clip off the carrier protein domain of EPO-A and express that on its own, and then enzymatically transfer an acetyl group onto that acyl carrier protein. And then that truncated acyl carrier protein could be used in combination with the non-ribosomal peptide synthetase to generate that methyl thiazole group that I showed in the previous slide. And I remember in my first group meeting, I presented these results, and Chris was very happy. And he said, all right, what are you going to do next? And I said, well, I have all these ideas for how I'm going to get EPO-A active, and I'm going to try expressing it under these conditions and those conditions, et cetera, et cetera. And Chris said, why? You have something that works. Focus on that. So this is the second rule of mechanistic enzymology. Actually, it goes beyond mechanistic enzymology, but in science, try to focus on what's working. And this is a lesson that's sometimes hard for me to follow, but I always try to remember Chris saying that to me, and focus on what works. And it's really harder than it sounds, but it's extraordinarily good advice. So I think I only really appreciated in retrospect after my postdoc Chris's really amazing history in the mechanistic enzymology of flavoenzymes. I mean, this is just a handful of papers that I kind of picked out yesterday that really highlight all of the beautiful mechanistic work that he did on this class of enzymes. And this really laid the foundations for an unbelievable number of discoveries in natural product biosynthesis. So again, I've just picked out a handful of natural product biosynthetic discoveries that were performed by Chris's group a little bit later. And all of these examples have beautiful flavin chemistry encoded with them. So it was really the early work in Chris's mechanistic enzymology in flavoenzymes that really set a very, very strong foundation for this later work in discovering all of these important natural products with pharmacological activity. And I thought for this talk that maybe I would be able to give a broad perspective and how some of Chris's early mechanistic work in flavoenzymes kind of, tied in and dovetailed in with some of these natural product biosynthetic discoveries that he made. But this is Chris. So Chris already did that. And I just really want to highlight your attention to this beautiful paper, which I reread. I'd read it a long time ago and then kind of forgotten about it, but I reread it yesterday. It's a really, really beautiful paper. Chris wrote it with Tim Wentzwick, his last postdoc, and this is where he really tied together all of the early work that he did with the mechanism of flavoenzymes and really showing case by case how flavoenzymes played into natural product biosynthetic pathways and really how the versatility of the flavoenzyme, the flavin cofactor could play a role in these biosynthetic reactions. So again, just some beautiful statements about how flavoenzymes work, and then tying it in with some of these details of all of these biosynthetic reactions that happen in the extraordinary chemistry of natural product biosynthesis. So I'm not going to attempt to recapitulate that because Chris did this far more elegantly than I would, but I highly recommend this paper. So with that, I really just wanted to say thank you, Chris, for teaching me all of this mechanistic enzymology and beyond. I also really wanted to thank all the Walsh Group alumni and collaborators from all the learning and wonderful times that I had with you. And I really want to thank the audience and the organizers for giving me the chance to give this really sort of brief and small perspective on Chris's work. And really, again, thank you for having me. And thank you, Chris.
Hi, can you hear me? Yes. Okay, hi everyone, I am Maria. I am a postdoc working with John Lom at Stanford in ChemH. And it is my pleasure to be here today presenting this very early paper on mechanistic enzymology from Christopher Walsh that deals with mechanistic studies on flavoenzyme L-alpha hydroxy acid oxidase from rat kidney. So this is a paper published in 1975 by Thomas Cromarty and Christopher Walsh. So as you know, enzymes are biological catalysts, and they are able to accelerate many chemical reactions. But sometimes enzymes on their own are not able to catalyze these reactions. So they need the help of an additional molecule called cofactor to help to carry out these specific reactions. So we therefore call apoenzyme to the protein fraction, which is non-active. And then once this apoenzyme binds to the specific cofactor, this becomes active, and then is able to metabolize a specific substrate into the product. And then in terms of cofactor, we can find many types of them. So we can find inorganic cofactors, such as metallic ions. And then we can also find many organic cofactors in nature, such as the flavins FAD or FMN. So to kind of give some context on what flavoenzymes are. So flavoenzyme is a term that we typically use to describe an enzyme that requires flavin as the cofactor. And this can be FAD or FMN, as I showed you before. So these flavoenzymes are very important oxidoreductase enzymes, and they are able to catalyze a wide variety of different reactions. So they are also extensively studied for these catalytic properties. And here I am just showing the general scheme for this flavin-dependent oxidation reaction. So you can see in the left the structure of the oxidized flavin, which is the active form of the flavin that is tightly bound to the enzyme to facilitate this oxidation reaction. So you can see how in this reaction, the substrate gets oxidized into the corresponding product. And then as a result, this flavin gets reduced, taking the protons and electrons for this electron transfer. And after that, this reduced flavin transfers these protons and electrons to the oxygen to generate, in this case, hydrogen peroxide. So here you can just see all the many chemical reactions that these flavoenzymes are able to catalyze. So as you can see, they participate in many processes in living organisms, and they are involved in many pathways like signaling, metabolism, or detoxification. But also flavoenzymes are very important in organic chemistry, given these unique catalytic properties that they have. And in all these cases, as you can see, that you can appreciate from this figure, the flavin is enabled this specific enzyme, flavoenzyme, to utilize oxygen in a very specific and dedicated and controlled pathway to carry out these very specific chemical reactions. So a useful tool to understand how these flavoenzymes work is to first understand which substrates they are able to utilize and how they utilize this substrate. So here I just show a very general scheme of how a flavoenzyme that is bound to the active form of the flavin, the oxidized FMN, is able to take a substrate and oxidize the substrate into the corresponding product. And this is a canonical pathway for most of the substrate for these flavoenzymes. But however, it has been also reported that sometimes when a flavoenzyme oxidizes a substrate into the corresponding product, this resulted oxidized product is able to somehow inhibit the activity of the enzyme. And this can be happening by affecting the cofactor, in this case FMN, or it can also interfere with the whole protein. So, and this is the kind of thinking that Christopher Walsh used to understand how this works. So he thought that if we break a process in which, in this case, it would be like this flavoenzyme catalysis, this way we would be able to understand how this whole process works. And this is the kind of logic that he used to understand all these mechanistics around flavoenzymes. And why they were so interested in understanding this mechanism for flavoenzymes? Well, by this time, so we are talking about 1975, a general accepted mechanism for these oxidations didn't exist. Well, it also doesn't exist nowadays, but by this time, the knowledge was much more limited around like, which is a mechanism, a specific mechanism for this oxidation. And also by this time, the resources were much more limited to study these flavoenzymes because you couldn't just simply buy or clone these proteins as we can do nowadays to study how they work. So therefore, at this time, they had to look for another resource of this protein, and they had to go to nature to look for these proteins. So this is a very clear example of this, the kind of research that was conducted at this time, which is the paper that Sarah presented before on the description of this rat kidney flavoenzyme. So in this paper, Thomas Cromarty and Christopher Walsh in 1975, which is just a little bit earlier than the paper that I am presenting today, they describe that this is a very unusual enzyme that has comparable oxidase activities towards hydroxy acids, but also amino acids. So as Sarah explained before, they are able to purify this enzyme from rat kidney, also determine the FMN concentration, and they also perform some preliminary studies with some substrates which are able to inhibit this enzyme. So this being said, this is a paper that I will be presenting today, which is a follow-up paper to the one that I just presented before. And this paper deals with the mechanistic studies of this previously isolated rat kidney flavoenzyme. So it was also published by Thomas Cromarty and Christopher Walsh in 1975. And they basically take this previously isolated flavoenzyme, and then they study which substrates can this flavoenzyme metabolize, and also which substrates can also inhibit this specific flavoenzyme, and they also determine by which mechanism this is happening. So here I'm just showing like a general overview of all the types of substrates that they explore in this paper. They basically focus on substrates that have been previously described to be substrates for other similar flavoenzymes. And these are beta-halo substrates, acetylene substrates, or vinylic substrates. So starting first with a beta-halo substrate, in this paper they're able to identify beta-chlorolactate acting as a substrate, a very good substrate for this enzyme. And this hasn't been reported before. And after that, they study the acetylenic substrates and the vinylic substrates. And what they observe in this case is that when the enzyme, the flavoenzyme, metabolizes these substrates into the corresponding keto product, they observe that the enzyme gets inhibited after this process. So in this paper, they are also able to determine by which mechanism these two different types of substrates inhibit the enzyme. And they determined that in the case of the acetylenic substrates, is by modifications in the FMN, and in the vinylic substrates is by modification on the enzyme. So I just wanted to show one of the main key experiments of this paper for you to get an idea of the type of quantitative measurements that Chris was performing to monitor this enzymatic reaction and to understand the mechanism of these reactions. And then how he was able to decode all this quantitative information to fully understand the mechanism of reaction for these flavoenzymes. So as I said before, they have previously observed that when they incubate the substrate 2-hydroxy-3-butanoate with the flavoenzyme, the resulting oxidized product is somehow able to inhibit the enzyme, but they still don't know how. So they decided to perform this time course experiment in which they monitor this oxidation of the substrate and then inactivation of the enzyme resulting after this process. And to be able to monitor this process, they couple this oxidation reaction of the substrate with the reduction of a dye, which is called DCIP, with the structure that you can see here. So the whole thinking of this experiment is that the enzyme oxidizes the product, the substrate, into the corresponding product. And then these 2 protons and 2 electrons from the reduced flavin will go to the oxidized DCIP that has color, but once accepts these protons and electrons, loses the color, and therefore we don't see this color anymore. So he would couple all these 2 processes to be able to monitor this specific reaction, so that when the enzyme is working, we see reduction of the DCIP, but once the enzyme gets inhibited by this specific molecule, we don't see any further reduction of DCIP. So then he monitored all these quantitative measurements in the graph that I am showing here. So if you focus on curve A, what he did was just incubating the DCIP dye with the substrate for the enzyme, and then he added specific amounts of enzyme at specific time points. So as you can see in curve A, when he added a 34 microgram of enzyme, this resulted in a corresponding amount of DCIP reduced. And then in curve B, when he doubles the amount of enzyme, he also sees a double amount of reduced DCIP. And he also observes that at some point, this reaction stops, meaning that the enzyme has been inhibited. So after the reaction in B stops, if he added more enzyme, you again see more reduction of DCIP to some extent. So this is very useful, and this way he was able to quantify this process, but besides that, he was also very interested in understanding what is the mechanism by which this oxidized product is able to inhibit this enzyme. So to figure out which was this mechanism, he performed additional experiments in which he measured which are the spectroscopic changes happening before and after we incubate the enzyme with this substrate. So here you can just see these spectroscopic measurements. So in curve A, you can see the spectrum of the native enzyme before it gets incubated with the substrate. So you can see the classical peak of the oxidized flavin appearing at 450, which is very typical. But then after they incubate this enzyme with the substrate, at some point the enzyme gets inhibited. And when they do another spectrum, as you can appreciate in curve B, they will say a bleach signal for the oxidized flavin. So the flavin is not active anymore. And then from here, they infer this postulated mechanism in which they say that this oxidation of the 2-hydroxybutanoate product leads to this corresponding keto product that is somehow inhibiting the enzyme by doing modifications on the cofactor FMN. And just to summarize a little bit all this talk, I hope that you kept the main key points. The flavoenzymes are very special catalysts in biology, and they participate in many different pathways, like signaling pathways, detoxification, and metabolism. Also, the cofactors such as FMN, these flavins play a key role helping these flavoenzymes to carry out these very specific and controlled chemical reactions. And finally, that we can use all this knowledge about the substrate and inhibitors to figure out what is the mechanism of reaction of these flavoenzymes. And we can also determine by which mechanisms these inhibitors are affecting the activity of the enzyme. So I would just like to thank all members of my lab, collaborators, and you for your attention and the opportunity. Now I will be happy to take questions.
So we don't have the privilege of asking Sarah questions, but I'm sure Lola would be happy to answer any of your questions on Walsh's first paper on a flavoenzyme. David.
So I guess for the mechanism where the butane is modifying the FMN, you should be able to come in with more FMN and reactivate the enzyme. Has anybody done that?
So yeah, they actually are able to identify like some kind of modification that is performed on the FMN in this sense. So it's just no longer active for this specific enzyme. Yeah. So yeah, they give like some key ideas on which could be this mechanism, and it's basically an addition of a molecule to the FMN. So you can no longer recover the activity of this FMN, and then the enzyme doesn't work anymore. Yeah.
I have a question.
Yeah.
Did you get from the paper what motivated Chris to use DSIP?
I guess it's just a typical dye that was used in those days. I actually, during my PhD, was also monitoring this reaction, and so it's still used nowadays to monitor this. So it's actually very interesting. Yeah, I guess it's just used as a second electron acceptor, really used in these reactions, and really useful to be able to quantify these specific reactions. Yeah.
Other questions for Lola? Thank you again.
Thank you.
So we have an opportunity for another short break. We'll keep it to no more than 5 minutes, so we can get started with the next session in time.
We actually are on time, and so we have until 11:15 until the next 2 rounds of speakers before lunch. So you have 20 minutes. Thank you, everyone. I think it's just a dam. I mean, for them to use. Yeah, the range. We have about 5 more minutes. 5 more minutes.
It is. Okay. Yeah, I do. I hope everybody is enjoying this teaching as much as I am. I know Olivia Fishbach, sadly in the first row, is listening with rapt attention and enjoying every bit of it, and I hope you are too. So, in the spirit of this event being for you, the Walsh trainee, the next session is essentially a Rorschachkian reflection of who Chris was. Chris, I know from my 30+ years of knowing Chris, loved every one of you, respected every one of you, knew every one of you better than you do, and wasn't shy about telling people like me about your amazing accomplishments, not just while you were with him, but also in the years after you went off and did all the great things that you've done with your life. So Chris everybody was Chris's favorite in the world of his alumni. However, in contrast to his trainees, Chris was not shy about demonstrating extreme favoritism when it came to molecules. And this session is a reflection of that. As Beth Sadley once told me, toward the end of his life, he actually also started to show the same kind of extreme biased favoritism towards atoms. And the whole series of books that you see, the order in which they came, sulfur came before nitrogen, and reveals something about a sense of Chris's bias. So clearly, there is a bias. And so the next session is really a preceptor and trainee pair who will tell you about aspects of this bias. So without further ado, let me welcome Yi Tang, a long-standing collaborator of Chris's, as well as a co-author on many of Chris's books.
Thank you very much, Chayden. It's truly an honor to be here. Can we get the slides? Oh, okay, it's my hand. Okay. All right, so I'm here to tell you about two subjects. I'm actually tasked to talk about many things, but the title I picked is Generating Complexity and Writing Books with Chris. So first, I have to tell you that I'm not a Walsh alumnus, unlike all the other speakers here. I actually worked for Chayden during my days at Stanford in 2002. And before I joined Chayden's lab, I read all his papers, every single one of them. And one paper in particular, a perspective in Journal Organic Chemistry that caught my attention, in which he talked about how during his postdoc in the John Innes, he met this great person, David Kane, who's sitting in the audience, and that really changed his career after he became independent at Stanford. And I thought, perhaps during my time at Chayden's lab, I could also meet this kind of person. And of course, I'd never imagined one of such person will be Chris Walsh, who became a very good friend and a very close collaborator. I met Chris through these famous Kosler, Walsh, Kane group meetings, and being somebody who's really a nobody at all, Chris was very kind to me, and from there we established a working and a relationship and a friendship. After I left Chayden's lab, Chris's lab and my lab collaborated on several projects, especially on fungal natural product biosynthesis. Shown here is one example on a molecule that we worked on, echinocandin, which is a glucan synthase inhibitor. Chris was really interested in this molecule because he helped push a derivative of this molecule into the clinic. And he also enjoyed working on this molecule because it has a lot of nitrogens and it's peptide-based. And I'll tell you about another molecule that we worked on later on in this talk. So when I was asked to give this talk, Beth first asked me to talk about the periodic table with Chris. And then the topic evolved into chemical building blocks. So I wasn't really sure what to talk about. But then I remembered, and also I didn't work with Chris, so I can't tell you all these things that Emily and Sarah told you about. Then I remembered sitting in 2013 Gordon Research Conference on enzyme and cofactors. Right before he retired, Chris entertained the audience with a top 10 list of his enzymes. So I thought this could be a great occasion to recap this list, and especially for the young researchers in the audience that you might not know some of these accomplishments by Chris. Some of these enzymes will already overlap with the previous talks, and it will overlap with the later talks, which I'll go over very briefly. So the number 10 enzyme on this list is the phosphopentothene transferase. Emily already talked about. This enzyme is essential in the post-translational modification of carrier proteins in both polyketide and non-ribosomal peptide synthesis. The next enzyme is this nickel-iron dinuclear hydrogenase involved in methanogen bacteria in the conversion of carbon dioxide into methane. So in this multi-enzyme process, the cofactor F420 provides reducing equivalence in 2 of the steps. And in order to regenerate the cofactor, a hydrogenase is involved using electrons coming from hydrogen. So Chris's lab was able to isolate this multi-unit complex by this raw figure shown here, and in which he showed there are 3 subunits involved with this enzyme. And the function of the 3 subunits were worked out much later by many researchers subsequently. A quick word on F420. This is something that I think we can appreciate Chris's love for flavin, even those that are actually not flavin. F420 is 5-deazoflavin. Compared to flavin, it doesn't have the 5-position nitrogen, which renders this molecule unable to do 1-electron chemistry, but still a very good hydride donor acceptor similar to the nicotinamides in NADPH and NADH. Chris and others had proposed that this enzyme, this cofactor, is involved in the last step of tetracycline biosynthesis by reducing the precursor into the active antibiotic. And my lab were able to biochemically confirm that 30 years later. But it's also this molecule that we can appreciate Chris's ability to simplify these complex biochemistry into things that we can all understand. For example, he said F420 is nicotinamide in flavin's closing, which vividly captures the structure and also the function of this F420. And he also said this F420 is more than evolutionary cul-de-sac. I think that's something only Chris can be able to describe in such detail. And those analogies and simplifications can be seen throughout his books and his writings. The next enzyme is a very interesting enzyme that it's a mercury lyase. It reduces mercury 2+ down to mercury 0, which is much less toxic and volatile. Chris demonstrated that this enzyme has a pair of active site cysteines that coordinate to the mercury 2+ and reduces to mercury 0. And its function is very similar to glutathione reductase. And what really struck me about this paper is this table here. You can see the detail that goes into a table that compares these similar enzymes in every aspect of biochemical feature you can imagine. And this is in 1982. And I think this kind of table is much more useful than most of the things we can find nowadays in publications. The next enzyme that is the D-lactate synthetase. This is an enzyme involved in vancomycin resistance, and you'll hear a lot more about this from Professor Jerry Wright's talk this afternoon. The next enzyme is the topic of Antonio's talk immediately after my talk. This is an enzyme involved in microcene C7 maturation towards antibiotic, towards becoming an antibiotic. This enzyme performs nitrogen-phosphorus bond formation. You'll hear a lot more in the next talk. The next enzyme is the enzyme that Emily already talked to you about. It's the cyclohexanone monooxygenase. This is one of my favorite enzymes. It utilizes the ability of flavin peroxyl species to oxygenate cyclohexane into the lactone. And this established that the Baeyer-Villiger mechanism is also present in enzyme chemistry. The next enzyme Emily also mentioned is a set of enzymes that incorporate dihydroxy benzoic acid into the molecule enterobactin, which is a very powerful siderophore to scavenge iron 3. And the next enzyme, number 3 now, is according to Chris, is the non-heme iron and alpha-ketoglutarate dependent halogenase that was also briefly mentioned by Emily, and you'll hear a lot more this afternoon as well. So now to the top 2. Both enzymes in the top 2 were discovered by Chris later in his career when he was working on complexity generation in natural products. The second enzyme is this aza-deoxyautomerase TCLM that's involved in thiopeptide maturation. So this is an enzyme that works on a ribosomally derived peptide that has already undergone significant oxidation and rearrangement to form these thiazoles. What this enzyme does is do a intramolecular azadeoxyl cyclization followed by aromatization to give the pyridine core of the thiopeptides. This large family is now known as RIPS, and Chris dedicated a whole chapter in the newest version of the Natural Product book to this family. So the last enzyme on this list, surprisingly to me and maybe to many, is this enzyme called asparlysin biosynthetic enzyme. This enzyme was actually a collaboration between my lab and Chris, so I was very happy when Chris said this is #1 during the GRC meeting. And maybe this is why I remember this list so well. So this enzyme is a non-ribosomal peptide synthetase, and it uses anthraquinolate as a building block in generating complex fungal indole alkaloids. So one of the themes for my session is chemical building blocks. I think anthraquinolate is a perfect example of the type of building block that Chris loves. This compound sits at the intersection of primary and secondary metabolism. It's multifunctional, it's cyclic, and most importantly, it has a amine group that Chris loves in molecules. This molecule is the precursor to tryptophan in primary metabolism, but it's also a key building block in many secondary metabolites, including the alkaloids that I'll be talking about, such as asparlysin. So these molecules are synthesized in fungi through a very concise biosynthetic pathway. And as Chris would often do, he would simplify these chemical logic and generalize them so that everybody can see it very quickly. The first enzyme in all these pathways is a non-ribosomal peptide synthetase that activates at least one anthraquinolate and at least one tryptophan. In the case of asparlysin on the very right, you see that 2 anthraquinolates are used. And this NRPS is able to leverage the unique reactivity and geometry of anthraquinolate to form these multicyclic structures very quickly. In the second level, Chris suggests that nature takes advantage of the indole reactivity and performs either a alkylation through prenylation or epoxidation by flavin-dependent enzyme to activate the indole ring for additional cyclization reactions. So as you see in the structure of asparlysin, this is a heptacyclic molecule that can be very quickly accessed by only 2 enzymes using anthraquinolate and tryptophan as the building blocks. Hence, this is the top 10 list by Chris, and you can see that his research interests really evolved from early on in the green, those in blue, in primary metabolism and also detoxification, to those in green that's involved in antibiotic biosynthesis and resistance, and lastly to the ones that involved in complexity generation. So next, I want to just shift gears and talk about writing books with Chris. It's really a privilege for me to be involved in a number of writing projects with Chris. As we all know, Chris is a prolific and often obsessive writer, and he wrote almost 1,000 papers, 11 books during his career. His 1979 book on enzyme mechanism is still a must-read for enzymologists today. And his book on antibiotics and post-translation modifications are Bibles for those of us who teach and study the subject. I was fortunate to write 3 books with Chris, 2 different versions of natural product biosynthesis and a vitamin book. And Chris also wrote 4 different books on elements and their roles in chemical biology. And as far as I know, Chris also has a number of monographs that was completely written but never published for different reasons. So I'd like to tell you about my experience with Chris and to show you some of the exchanges we had to show you how Chris motivates people like me and also how he thinks about the exercise of book writing. So between 2016 and 2018, we wrote, and we kept writing. We wrote 2 books that are both 700 pages long. We also wrote a chemical review article that involved 1,000 compounds. And we also wrote this review on molecules that power metabolism that was gently rejected by Joanne Stubbe because it was 3 times too long for the journal. And we also then wrote a 17-pager biochemistry article that I think might be the longest in biochemistry for a while. During COVID, we updated the first version of Natural Product and wrote the second version. So although these projects were really time-consuming, and I think Diana also told Chris that it's really taking too much of my time to do this, but these actually were the most productive years of my own career in my lab, because just think about it, you get somebody like Chris you can constantly talk to and give him information about your own research, and he gives you honest feedback and assessment. So it's really a privilege for me to be working with Chris during these times. And also really learned and appreciate Chris' work ethics. He wrote during family vacations at exotic places that he told me about. He wrote during preparation for his 50th wedding's anniversary. And also he wrote during very challenging health conditions at times. So to me, to be able to work with Chris during these times is truly an honor. So I was surprised that Chris picked me to write these books. I interacted with Chris a lot prior to that, especially during a Stanford-UCLA collaboration with Chris, an advisor. I often send Chris decks of slides and collection of papers that he's interested in. Maybe that's why. But Chris has a very interesting way of convincing co-authors to work with him, as you can see here in red. So he says that these books nobody reads books anymore. So he also said, this is not how I should spend my time. And number 3, that not clear any publisher will ever be interested. So that's in his invitation email, and he also told me to think long and hard about saying yes to him, and it can always change if I decide to. But I'm glad I accepted right away and began this project. Chris, throughout the process, Chris motivated me, and Chris also provided encouraging words to me because it was truly a grinding process with a book and a chemical review at the same time. So Chris would say, okay, you should go back to do your own things now, then follow up with a draft. I'd like to see some figures worked on this draft. And then Chris also said, this is less humorous, but also I think what he truly feels is that writing books is a continuing self-education process. And he felt during this book writing process, we learned a lot about these chemical reactions that organic chemists might never be able to appreciate. And there are some more humorous things, and he wrote a particular email, and I really don't understand what he was talking about. But I think I know that he thinks I just truly have the same energy as him. And then lastly, this email that really stood out in my memory is that he suggests me to do other things than doing this particular exercise with him. Anyway, it was a very enjoyable time, thousands of emails exchanged. And then to wrap up, and after that, we decided to write a couple of additional books and review papers on primary metabolism. And I think this is where, already referred to by Emily, on Chris's thinking about the role of these metabolites in function of a cell. They're kinetically stable but thermodynamically activated molecules. Chris initially said he wants to write about 6 molecules, but then after a little bit more discussion, we expand to 8 to include his favorite molecular oxygen in the list as well. So this was a very enjoyable review paper that we wrote. And then from there, we decided to write on the vitamins because these molecules extend perfectly into cofactor functions. And then Chris also offered his view and his normal invitation to do another book with him. So to conclude, it's really a privilege to work with Chris. I was not in his lab, but I feel like I learned so much from him. Working on these books is not just about writing. It's about education on anything I've ever experienced in my life. Chris taught me how to be a great scientist. He also taught me how to be a good scholar. Through his emails, I can see and I learned from him how to be a good husband, as well as a good father, and also to be a good person. I think I strive every day trying to be like Chris, but I know that I'll never come close to who Chris is. I miss him very much, and I wish I can still talk with him. So thank you very much.
So the trainee in this session will make Oedipus proud, because he's not only Yi's grandson, but he's also Yi's brother. So go figure. Antonio Del Rio Flores trained with Wenjun Zhang, who was a Chris Walsh alumnus when in her lab across the bay at Berkeley. And he is now a Stanford Science Fellow who is being hosted in my lab as well. So welcome.
Perfect. All right, hello everyone. Fantastic talk by Yi. I'm very grateful to be here today. Not only commemorate Chris's illustrious career as both a scientist, but also as a esteemed family member and a mentor within our community. And today I'll be focusing on, my opinion, one of Chris's most interesting papers in the field of heteroatom enzymology. When I started this PhD, I was really interested by this paper, and it really inspired my own work. And today we're going to be focusing on how the nitrogen-phosphorus bond is constructed on this small molecule called microcin C7, which is an antibacterial peptide that is produced by E. coli. And with that, I'd like to go ahead and get started. So let's start off by talking first about what are microcins. Well, microcins are a family of natural products that are ribosomally produced. And in general, they're quite small. They're less than 10 kilodaltons in size, and they're secreted by some enterobacteria, particularly E. coli. And what I want to point out here is that these molecules are used as defense mechanisms to be able to kill off bacterial competitors. Their MICs are in the low nanomolar range, so they're quite potent to be utilized. And what I want to also point out is that from a biosynthetic standpoint, a precursor peptide is produced by the ribosome, as I mentioned before, and they undergo these post-translational modifications that are key to impart all the various bioactivities that are present in these small molecules, and they also impart their physiological function. And what I want to also point out is after they're specifically produced is that they're exported from inside of the cell, and then they float around the environment until they reach a specific bacterial target, and that's when they import their specific bioactivity. And for today's talk, we're going to be focusing specifically on one small molecule from the microcin family called microcin C7. And I want to point your attention to the small molecule for a couple interesting key features. Number one, on the N-terminus, we have a formal methionine. And then if we look at the C-terminus, this is really where the importance comes in from the perspective of a warhead. We have this phosphoramidate linkage that links together a heptapeptide with AMP. And we observe post-translationally also a flip from an asparagine to an isoasparagine. And then lastly, we have this aminopropylation that happens on the phosphoramidate oxygen. But for today's talk, I want to focus on how this phosphoramidate linkage is being formed for a variety of different reasons. The first thing that I want to point out is that for several decades, we've used different types of phosphoramidate nucleoside analogs to treat a variety of different types of human health conditions because they serve as enzyme inhibitors. Some applications can be used for HIV, cancer, as well as COVID-19. So being able to understand the enzymological logic to be able to install these nitrogen-phosphorus bonds are key in many different regards. For one, it can help us design novel antibiotics, as well as being able to generate novel catalysts to be able to afford these different types of biochemical transformations. So we're going to be focusing on how this is formed in nature. Okay? But before we do, I want to tell you a little bit about what is microcin C7, what does it do from a biological perspective? And the way that I would like to paint this perspective is by telling you a little bit about what it's known as. It's known as a Trojan horse antibiotic. And the way that I'll explain what this means is by taking everyone in the room here and also virtually and taking you on a mini field trip. We're going to be going on a field trip to a producing cell of E. coli that generates microcin C7. We're going to go to the cytoplasm. And inside of the cytoplasm, what's going to end up happening is the ribosome is going to produce a precursor peptide, MCCA. This MCCA is going to undergo several post-translational modifications to generate microcin C7, which is the final decorated natural product that imparts the biological functions. Once this is produced, what's going to end up happening is there's going to be a transporter that's going to take the cytoplasmic small molecule and release it to the environment, and it's going to float around in the media, and then until it comes in contact with a specific transporter from a susceptible cell. And it's going to be end up taking up, and once it's inside, it gets a little bit more interesting. What's going to end up happening is that this small molecule is going to get chopped up by endogenous proteases. It's going to generate this small molecule called 1A. One of the structural features you can observe from compound 1A is that it looks a lot like an aspartyl AMP mimic. So as you would imagine, this small molecule inhibits the activity of aspartyl tRNA synthetase, which in turn blocks protein synthesis. And ultimately, this is just fascinating how this small molecule goes inside of target cells, and it undergoes this degradation pathway to be able to generate this really important type of bioactivity. And today, I will be focusing on how this nitrogen-phosphorus bond is indeed generated, because it is truly the main key player in this enzymatic transformation, because this phosphoramidate linkage, as we'll see later, is quite hydrolytically stable, and we'll be understanding how this is done in nature. Okay? So what Chris ended up doing, he started off his studies by first analyzing the biosynthetic gene cluster of microcin C7, which is right here. So in general, microcins in common have several commonalities within the BGC. There's a gene that usually codes for the structural precursor peptide, and there are also several other genes encoding proteins that are important for the maturation, immunity, and export of the final decorated compound. And ultimately, from his in silico analysis, he was able to predict the function of several of the encoded proteins. And what really caught his attention was this enzyme called MCCB, which was annotated as a homolog of an E1 ubiquitin ligase, and also a homolog of TF, which is an enzyme involved in thiamine pyrophosphate biosynthesis. And ultimately, his hypothesis moving forward was that this predicted adenylating enzyme would be responsible in generating the nitrogen-phosphorus bond linkage. And in order to test his hypothesis, he went through the steps of chemically synthesizing an analog of the precursor peptide MCCA. So one of the differences that you'll see here, if we look at the N-terminus, we don't have the formal group. He used a mimic that just has a free amine. But ultimately, what he was able to show is that if he incubates this substrate with MCCB that was purified from E. coli, along with co-incubation with ATP and magnesium, he took that enzymatic reaction, analyzed it with HPLC, and was able to arrive at a couple interesting key features. He was able to observe 3 new products that were formed in an ATP and magnesium-dependent fashion. And taking a closer look at these enzymatic reaction, if we look at this enzymatic reaction at absorbance at 260 nanometers, we can see that the putative peak that was annotated as AMP in compound 3 absorbed at 260, indicating that some type of adenosine moiety was present. And on the other hand, compound 4, which is the third product, it did not absorb, indicating that this adenosine moiety was not present. So at this point, what I want to bring up to everyone, we have these 3 new products, and we wanted to figure out which one, the identity of these 3 individual products. The first one that we can talk about is this peak that was putatively annotated as AMP. What Chris did is that he ran it in parallel with AMP and ADP standards. It turns out that this molecule co-elutes with AMP. And what this suggested is that the ATP utilized in this reaction was being hydrolyzed into pyrophosphate and AMP. Now, at this moment we can move on to the next product, which is compound 3. So compound 3 is quite interesting because it's the other species that absorbs at 260 nanometers. What Chris did is he compared the high resolution mass between the precursor peptide and the compound 3, and he found a 329 mass unit increase, indicative of an AMP ligation. So at that moment, he scaled up the enzymatic reaction and was able to structurally elucidate the identity of compound 3. And one of the things that you can note from compound 3, it looks very similar to microcin C7, apart from the formal group not being on an N-terminus. It's very similar with the exception of the aminopropylation on the oxygen on the phosphorus. So at this point, we have an explanation for 2 out of the 3 products. So the next part was to figure out what is the identity of compound 4. So again, by high-resolution mass, Chris had a hypothesis that perhaps this precursor peptide may undergo spontaneous cyclization to generate a succinimide, and this was consistent with the loss of 18 between compound 2 and 4. And this is something that happens quite commonly when synthesizing these peptides through solid phase synthesis. In order to test his hypothesis, he chemically synthesized this peptidyl succinimide compound 4, fed it into MCCB, and what he was able to find is that compound 4 was consumed by NCCB in an ATP and magnesium-dependent fashion to generate the phosphoraminate compound 3. And ultimately here, it also turned out that this compound 4 co-eluted with the enzymatic product. So at this point, this indicated that this succinimide was an intermediate en route to phosphoraminate biosynthesis. After performing some kinetic characterization, Chris was able to show that it was quite a catalytic competent intermediate with a catalytic efficiency that was about 2 times higher than the starting precursor peptide. And ultimately, it gave us an insight into perhaps what the reaction mechanism was. But before we jump into the mechanistic details, what I want to go over right now is to tie up some loose strings in the way that kind of Chris went through this process. One of the really interesting things about this enzymatic reaction, if we compare the structures of compounds 2 and 3, if we look at the C-terminus, we observe that initially we have an asparagine, and then with compound 3, we see the nitrogen kind of shifted away to an isoasparagine. So one of the things that he wanted to test was to see if the carboxymido nitrogen on the precursor peptide was retained in the enzymatic reaction. So he prepared a 15N labeled version of the substrate, fed it into the enzyme, and he was able to analyze the labeled version of compound through high-resolution mass spectrometry, and to be able to see a 329 mass increase consistent with the unlabeled case. And ultimately, what this indicated was that both nitrogens from the asparagine were incorporated into the final molecule compound 3. The second thing that I want to point out before we move into the mechanistic details is that we showed previously that ATP is required to convert the precursor peptide into compound 3. Same case with the succinimide. And what this would implicate is that 2 ATPs would be required for this enzymatic transformation. And ultimately what Chris was able to show by performing biochemical assays with varying degrees of ATP, that he observed complete conversion of the precursor peptide when 2 or more equivalents of ATP were utilized in corroboration with his hypothesis. But ultimately now we ran into the question, how was this nitrogen-phosphorus bond formed, knowing now that we have the succinimide as an intermediate to that final product, compound 3? And the way that he dissected this problem was actually quite interesting. So we know compound 4 is an intermediate. We want to get to compound 3. If we compare these two structures, there are 2 different events that must happen. Number 1, we need to ligate AMP to the C-terminus. And 2, we need to find a way to break apart the succinimide ring. If we're able to do that, then we can propose a mechanism to be able to get to the final product. And based off of this, there are 2 different potential pathways that this can happen. First, we can go through a ring opening process followed by nucleophilic attack of ATP, or we can go the other way around. It could turn out the succinimide acts as a nucleophile to activate ATP, and then followed by ring opening to generate this molecule. So Chris's strategy at this point was very clever. He thought that if he can chemically synthesize one of these potentially intermediates and test it for an enzymatic reaction, he can use that to discern between the 2 mechanisms. So he decided to chemically synthesize this isoasparagine heptapeptide. And we'll talk about that in the next slide. So this molecule, the same one from the previous slide, was chemically synthesized, fed into the NCCB catalyzed reaction, and ultimately from a time course analysis, what he was able to observe that it was not converted into compound 3. But interestingly enough, if he performed biochemical assays with the precursor peptide and spiked in different amounts of this isoasparagine heptapeptide, he observed a dose-dependent inhibition of this final molecule, indicating that this isoasparagine is not an intermediate, but actually serves as an inhibitor of this enzymatic reaction. And what does this mean for us from a mechanistic standpoint? Well, this means that this at least suggests that the succinimide has to perform a nucleophilic attack of the ATP before the ring opening process happens. And based off of this now, I want to end today's talk by mentioning his final proposed mechanism and give some insights into what is happening here with this enzymatic reaction that I find just quite fascinating. Okay, so there's a lot of chemistry that's been going on, but I kind of want to simplify this, be as succinct as possible. We start off with our precursor peptide. We have the carboxylate attack the first ATP molecule to generate a hydrolytically labile AMP intermediate. What happens is that the carboxymido nitrogen attacks the carbonyl carbon to release AMP and also generate the peptidyl succinamido group. Compound 4, again, as we mentioned before, was an intermediate en route to compound 3 formation. So at this point, we've got through half of the mechanism. This compound 4 then attacks the second equivalent of ATP, and then it generates a phosphoramidite intermediate that lends itself to an attack by water to catalyze the ring opening process to finally generate compound 3, which is the final product from this enzymatic reaction. And ultimately, this mechanism is consistent with several of the key features that Chris highlighted in his work. Number 1, that 2 ATP equivalents are required for complete conversion of the precursor peptide, and number 2, it involves the formation and breakdown of the succinamide intermediate. And then lastly, what I want to just briefly mention is really some of the fascinating things that I think are happening in this reaction. So one of the things that I think is a remarkable feat of enzymology is that ATP is utilized in 2 different ways in this enzymatic transformation, and these two different pathways have completely different outcomes. The first one, if we look at the carboxylate activating ATP, this is what commonly adenylating enzymes do. They generate a hydrolytically labile intermediate that usually lends itself to attack from some type of nucleophile. But really where the part that I became very interested from this paper was that this succinamide intermediate compound 4 attacks the ATP and generates a hydrolytically stable intermediate, something that remains stable as you would expect, and it's responsible for generating the warhead that needs to remain intact inside of the cytoplasm as it's being exported into the environment and it's imported into the target cell to impart its mode of action. And ultimately, that's the main reason why this is a very interesting enzymatic transformation that is happening. And with that, I'd like to end with just some acknowledgments. Of course, I'd like to thank Chris for being an inspiration through generations of scientists, and his work will leave an everlasting impression to several generations until the end of time. I'd like to also thank Beth and the organizing committee for all their hard work and being able to put together this teach-in, as well as to thank my academic grandfather and I guess brother now, and also my postdoctoral and also doctoral advisors. And I'd like to thank everyone today for coming today and allowing me to share my story today.
We have time for either conversations with Yi or questions for Antonio. Tim.
Hi. Very cool talk. Thanks.
Who's this for?
This is for Antonio.
Okay.
Right at the end, you had compound 6. The succinamide is not symmetrical. Is there, I mean, empirically, observationally, we see which one gets formed, but is there a reason for attack on one versus the other?
Right. So your question is, let's go back. Yeah. So you're saying, yeah, no, that's really interesting. So one of the perspectives here is really the site-directed like hydrolysis on one side of the other side. But that's something that I guess has not been really hashed out. Equivalently, it could be done from either side, but at least if it goes through that side, it is consistent with the final structure of compound 3. But I agree with you. This is one of the reasons why enzymes are so interesting, being able to have these transformations happen on a very regioselective area, as opposed to one that is also chemically also equivalent.
Another question?
Nice talk, Antonio. I guess my question's for Antonio as well.
Yeah.
Following up on that point, I guess, were they able to show in this paper, do they know, does that final hydrolysis step of 623, does that happen in the enzyme active site, or does the 6 diffuse away and then water adds in?
That's a great question. So from my reading, there was a follow-up paper in which they solved the structure of MCCB both in its apo form along with several different types of substrate-bound structures, both with inhibitors and intermediates. And what they proposed in that paper that everything was done within the active site.
Thanks so much, both of you. My question is for Yi. As someone who's worked so closely with Chris, especially on the writing, what I've always been impressed by is just like the volume, just the sheer volume of his writing.
Yes, I agree.
Could you please share with us, as someone who's now worked with him, who worked with him for many years, how did that volume come to be?
As I mentioned, Chris, I think he just loves to write. I visited his Cape Cod home where he has a desk that's confined between two walls, and he said once he's in the mode sitting at that desk, he just does nothing but write. He can just do that for hours. But he can do that for hours anywhere. I think, as I mentioned, when he's on trips in Europe, I think it was Czechoslovakia or one of the countries, I forgot which name of the country, and he just loves to write. And he often say, okay, I'll give you something, let me think about it, and then half an hour later he'll give me 3 pages of or something that to digest. So yeah, Diana.
So I have to jump in.
Yeah.
So I was married to him. For 57 years. And he did love to write, and he wrote all the time, and all my images of him pretty much are writing, but he was a very, very, very present husband and father. It was amazing. He managed to do them both. I mean, to be so very prolific and to be there for me whenever I needed him, no matter what. I want to tell, and there was a point when he was sick, this was, I think, before you started collaborating, and he told me, he had this autoimmune disease, and he said to me, if I die and the book isn't done, the person you should get in touch with is Yi Tang. There was a book he was So he knew, he had great faith in you. There was a great little story, I'll be quick. So there is a professorship now for Chris at Harvard Medical School, and the first holder of the chair is a man named John Clardy, who's, probably you know his work. And so there was a special event to inaugurate John as the first holder of the Chris Walsh professorship, and John told this story about being at it wasn't a Gordon conference, it was a special conference of the great poobahs of chemical biology, and John sort of telling in this kind of homespun way, and so he and Chris are there, and it's some foreign country, and it was a big, important conference. At the end, the sponsor said to John and to Chris, I want you two to go and write this up, please, because this is important for the field. So they say, okay, but it was apparently sort of not very clear what to write up. So Chris went home and wrote it up, because that's what Chris did. And he sent the copy to John, and John didn't know him very well at that point. He was at Harvard, he'd moved to Harvard from Cornell, but he didn't know Chris that well. So he gets this email from Chris Walsh, and it's the write-up, and it's pretty much all there. It's sort of halfway down the 5th page, it says, Clardy, fill in this point. And then halfway down the 10th page, it says, Clardy, say something here. But other than that, it's kind of done. So John ignores it for a while, because he doesn't know what to do about the things he has to fill in. And so Chris sends an email, Clardy, have you done it? No, John hasn't done it. And finally, Chris sends an email. Oh, no, they meet over lunch, and Chris says to John, John, do you know that I write books? And John says, well, someone mentioned that to me, that you write books. And Chris says, do you know that I'm usually the first author of the only author of the books? And John says, no, yeah, I guess I did notice that you were. And Chris said, well, now you know why. So this is the guy.
Thank you. Great job.
On that note, how can you top up that, that vignette? So we have a lunch hour now, and I hope you continue connecting and enjoying the Walsh memories, which I'm sure are in abundance in this room today.
Awesome. Thank you, everyone. Can we give a round of applause for the first session? We also have lunch now outside of the doors. You could pick it up. You could eat anywhere on the inside. If you keep going outside down that way, there's tables outside next to the fountain. Hopefully it's not as cold as this morning outside. But thank you, everyone, and we'll be back at around 1 o'clock, I believe. We'll make announcements moving through the building as time gets closer. Thank you.
Size exclusion and sucrose sedimentation. So I was now a year into my postdoc with nothing to show.
everybody. I'm Jerry Wright. I'm at the McMaster University here up in
Okay, hi everyone. We're gonna get started soon for our second session. Just some housekeeping announcements. If there is still food outside, feel free to grab more food to take for later. Additionally, during our second break, which starts at 2:30 p.m., if you are a Walsh alumni, we are going to try to take a group photo here on stage. So after this session, if you are a Walsh alumni, please, during break, come up on stage so we could take a group photo. And if you know of any other Walsh alumni that are here in person that you don't see around, please send them a text message or email or however you get in contact with them to be here at 2:30 if they want to be part of that group photo. Including undergraduates who were in the Walsh lab. So if you in any context were mentored by Christopher, please be here 2:30 after this session during the break for a group photo. And next for our moderator, we have Michael Fishback, who will introduce our speakers.
Thank you, Bria. Okay, this next session is going to be a lot of fun because it has to do with a topic that became an obsession of Chris's in the second half of his career, and that is these giant enzymes that function as assembly lines to build giant molecules that are famous because they've been used frequently in the clinic, and they go by the technical term polyketides and non-ribosomal peptides. Chris became fascinated with the logic by which these enzymes worked, and this was also the topic that led him into a long-standing collaboration with David Kane and Chetan Khosla, who are both here, and Mohammed Marahil, who is not. There are really two big question areas when studying these enzymes, and the first is how do they function? How do they pass substrates along the chain to make a big loop or a big linear molecule? And then the second question is, how are those molecules decorated after the fact? Because they're never just left alone. There are always additional chemical groups put on. And we're going to take those up in reverse order. So first we're going to talk about a decoration, and then we're going to talk about the logic by which these enzymes act. And so next two speakers. First is Fred Valancourt, who's the VP of Biomolecular Sciences and Lead Discovery at Remex, which is a Boston area biotech company. He was a postdoc in Chris's lab around the same time I was, from 2003 to 2006, where he, along with Ellen Ye, who's in the audience here, broke open an area that was something that Chris was fond of dearly, which is the way that enzymes add halogen atoms to natural products, something that was nearly impossible to imagine chemically, but enzymatically was difficult to study initially and then broken open by Fred. He is accompanied by Kate Bauman, who's a postdoc in my lab, studying skin bacteria and how they could be used as vaccines. And she was a graduate student before that with Brad Moore at UCSD, where she studied similarly capable and mysterious enzymes that were involved in the biosynthesis of another natural product that's used as a drug called selenosporamide. So I introduce Fred and Kate, and I note for you that this session's going to be a little different from the last one in that the presentations are not going to be in sequence, they're going to be tag teams. So we'll see how that format works for introducing the topic.
Thank you, Michael, for the kind introduction. So we'll dive right in, but before we get into the halogenases, we'll have a brief intro on the tailoring enzyme as a whole class at first. Chris, in the late 90s, early 2000s, as he was really getting back into full-fledged natural product biosynthesis discovery, got really interested in the whole machinery that we'll hear next, but also got really involved in the tailoring enzyme themselves, as can be seen from this 2002 review that was written at the time that really became a really big source of ideas for other labs as well in parallel to this. And just turns out that just very recently, it's one of the latest review that he wrote that was published earlier this year. It's over 60 pages of material in there that came out earlier this year. So we're not going to go over this again, because we just can't do justice the way that he can. But in that most recent one, he's reviewing all the material since that initial review. And along those years, there were also several other kind of smaller but still very impactful reviews that were written together with many other focused articles on different classes. It was just amazing at writing, as we heard before. Here's an example of different classes of tailoring reaction, right? Methylation, glycosylation, cyclization. I'm sure we'll hear some in the next talk from Raul a little bit. Oxidation and halogenation, which is more the focus of this talk today. And I just want to show some figures from the 2002 review that really highlight how, they're a bit fuzzy, but really highlight how Chris was tackling the problem, right, or the intricate biology at play, in which he would basically take a sheet of paper and kind of draw the mechanism, and the goal was always to show as simplistic as possible how the electrons are moving around, right, really mechanistic enzymology at play. On such complex protein machinery, right, these enzymes are hundreds of like kilodalton big, so it's quite an adventure. Another area that he was heavily involved in, there are several seminal papers on this, is characterizing epimerization, in which you could move from L-amino acid to D-amino acid, right? A lot of people would kind of just skim over this in natural product, but Chris really wanted, no stone is left unturned, right? To really want to characterize that. But if you think about this, epimerization is a different problem to tackle because there's no mass change. There's not much changes, right? So you need to move to chiral columns and all that to characterize that MOA. So that, and he did all the way. And I'm sure we'll hear more, as I said earlier, but cyclization, which is really an intriguing tailoring enzyme class in which some domains embedded in those mega synthetase could cyclize the whole thing, as well as for the epimerase and things like that. These are embedded domains. There are also several other genes within these clusters that are not always embedded in the mega synthetase. And that's from the most recent review, where Chris had always had the amazing talent to really simplify things at the highest level and really classify and bin things in reaction types, right? And that's, to me, one of the greatest example of it, where he could categorize the enzyme by group transfer or oxidative in his most recent review. And you can see how the mechanistic is simplified to very, just a few bonds and a few electrons moving around. So that brings us to the halogenases that Kate will introduce.
Awesome. Okay, so it's my job today to introduce you to the decoration of choice, which in this case is halogenation. And when we talk about halogenation of a natural product, what we mean is the incorporation of, you can see this group on the periodic table, this column that I've highlighted in orange, the incorporation of a halogen atom into the final structure of the natural product. There are thousands of known natural products that are halogenated, and most commonly we see the incorporation of chlorine and bromine. So you might be wondering, why do we care about halogenation as a tailoring reaction in natural product biosynthesis? And if we think about it from a pharmaceutical perspective, it's because halogenation is often seen really commonly in clinically used drugs. So I've put a few examples on the slide here of drugs you might be familiar with, but you might not realize that they're all halogenated. And that's because the inclusion of a halogen in a molecule can be really important for enhancing certain pharmacological properties. So it can enhance the lipophilicity or permeability of a molecule. It can also enhance small molecule binding to its protein target. And furthermore, the halogen bond is highly analogous to a hydrogen bond. And so you can include a halogen in a molecule without interrupting any other important structural features. And so while we know that halogenation is used commonly in pharmaceuticals, it's something that nature does really well as well. So I'm showing a few examples here of natural products that are halogenated, and there's three things that I want you to take away from this slide. So one is the diversity of bioactivities that you see of these molecules. So you can see that they range from hormones to antibiotics to anti-cancer compounds. The second is the diversity of source organisms. So halogenated natural products are made across the tree of life. So you can see we have compounds from mammals, like our own thyroid hormone, thyroxine, to compounds produced by marine bacteria. And then third, I want you to note the diversity of the site of halogenation in these molecules. So these molecules are all structurally very different, and you can see that the site in the molecule that's halogenated varies across all of these molecules. And so halogenation in nature is often very important to the bioactivity of a molecule. And so an example that I'm illustrating that with here is this molecule, Selenosporamide A, which is produced by a marine bacteria. It's a potent and irreversible inhibitor of the proteasome. And because of that bioactivity, it's actually currently in phase 3 clinical trials for glioblastoma, which is a rare and deadly form of brain cancer. And that ability to inhibit the proteasome irreversibly is really due to the presence of this chlorine. So I'm illustrating the mechanism of action at the top of the slide, and you can see that there's a covalent adduct formed between the proteasome and Selenosporamide A thanks to that beta-lactone moiety. But then it's displacement of that chloride and subsequent formation of the THF ring that renders Selenosporamide A irreversibly bound in the active site of the proteasome. And when you look at non-chlorinated analogs of Selenosporamide A, these molecules, which are naturally isolated from the environment as well, they lose a lot of their bioactivity with the loss of that halogen. And so because of all these reasons, these are all, we know that halogens are important for pharmacological properties, they're important for the bioactivity of molecules, and so this is of course why this is an interesting decoration and why Chris was interested in it. So Fred's gonna tell you a little bit about some of the enzymes that are responsible for installing halogens.
So back in the early 2000s, Chris binned the different halogenases in these various classes, right? Class 1, the alloperoxidases, was well known and reasonably well characterized, right? He kind of never really got into those. But then for class 2 and 3, these were really the novel class that he got into. And we heard a lot about flavin already, and you know, he wasn't done with flavin, right? Like when he could see analogies in natural product gene clusters that were getting sequenced at that time with known flavin oxygenase. It was like, oh, what if, you know, they're actually doing the halogenation reaction, right? Like that's really, you know, the creative thinking that he had, you know, all along over this. And the same was true for other natural product biosynthetic clusters in which we're seeing homologies with non-heme iron oxygenases from the alpha-ketoglutarate family. And these were halogenating carbons that are quite inert, right? They're sp3 carbons like in the syringomycin E, whereas the flavin oxygenase are more involved in activating halogenating electron-rich saturated carbons. In addition, there are also different natural products that are cyclopropane derived as the end product, in which there is a chlorinated intermediate that leads to the final product. And basically, back in those days, Chris tackled not one enzyme, right, but 5 of those, you know, almost upfront and went at it and characterized those. So there's the SIRB2, right, one of the now most well-characterized non-heme iron halogenase, together with two other ones that are involved in coronamic acid or ACC biosynthesis. And in parallel to this, he was also tackling, you know, the intriguing flavin class that involved in rebecamycin and pyroluteorin biosynthesis. And how did he get into this, right? So, you know, no stone went unturned. Obviously, it goes without saying that it's a very interesting biological problem to tackle. How is this halogenation happening? And that was really the laser focus, right? What's the mechanism leading to this? So all this creative thinking and what I said, you know, about finding genes that could potentially do the role in these various biosynthetic pathways was really what got this going. And think of this was the early 2000s. So when you saw a gene in a cluster, you needed to get the gene from the source. So it was either getting, you know, the bacterial strain, which he successfully did for rebecamycin, or finding creative ways to get to other gene clusters. Turns out that for the coronamic acid article, right, we didn't go with that strain, but there was another plant pathovar, so that's a plant toxin. There was another plant pathovar, like sequence genome, that had just been deposited in the database, and that strain was available from ATCC. So basically, purchase the strain, clone out the gene, old school, you know, restriction enzyme, and get going at expression and going at it. Just a few years in, the postdoc, you know, things change. Synthetic DNA was becoming more common, and we're like, you know, Chris, we probably can stop doing that. There's a company in California called DNA 2.0 that are getting pretty good at making genes. So when we got into barbamide synthesis, trichlorinating enzyme, we just could order the genes from the sequence found in the database, and that became kind of the new motto. But back then, Chris spent a lot of time writing emails trying to get the genomic source for getting at all these intricate pathways. So here, highlighting the flavin class of halogenase. That was really an effort driven by Helen Ye, that is in the audience, really tackling both ribacomycin and pyruvate synthesis using quite a reactive intermediate, and always thinking, there's probably a direct chlorine intermediate with the flavin itself. They characterize the biosynthesis like exquisitely, that led to two very nice PNAS papers on both enzymes. The one on ribacomycin is cited here. The main way that these pioneering papers were put together was using radioactive intermediate, right? This is like old school, like back in the days when Krebs did the cycle, the citrate cycle. You just isolate intermediate and figure out what they are, and that's basically how Chris tackled the problem, using either tritiated or C14 labeled amino acid. But Chris had this idea that chlorine can also be radioactive. Like, I'm not sure, a lot of us before these papers came out knew that. So we just purchased, radioactive sodium chloride, right, table salt, that's radioactive, and then incubate with these various enzymes and could detect, radioactivity that could show chlorination unequivocally. And we could even do this on the protein gels as well to show that it was covalent on the substrate and all that. So that was exquisite. Then he teamed up with Kathy Drennan at MIT to try to crystallize these things, and we'll go into details on this later. But one thing for the flavin class is once they were incubating with the radioactive chloride ion, you could desolve the protein, and the protein was still radioactive and it could be stable for like 60 hours. So then it was like, oh, we really made a cool discovery here, and they basically, started drafting Science and Nature draft, to really, because this was really high impact. But turns out that isolating that intermediate became extremely challenging because probably within the active site, it's a chloramine intermediate on the lysine, is stabilized in some way. And as soon as you try to characterize it by, denaturing the enzyme, it just too labile and it goes away. Having said that, this enzyme could be crystallized, and it crystallized rather quickly in the wild type form. Sure enough, when it's, the modified form, it took 2 weeks for the crystals to grow, and then given the half-life, the intermediate was gone. So that was, I'm sure, a rather frustrating endeavor. But nevertheless, they were able to get it published into biochemistry, and that's kind of a paper that is referred a lot. And in a way, some people agree with the idea, some not, but at least the idea is out there. And I think we're quite convinced there is definitely a stable intermediate in there. And as Ellen was telling me earlier at the break, Chris was always extremely precise in the way he writes things, and that's exactly what the title says. Long-lived intermediate does exist, and that's definitely true, right? What is it? Is it the chloramine, or is it something else, right? Someone's, probably will identify it later on, and I bet will say Chris was right. So with that, let's move on to the other class.
Okay. So Fred just introduced us to this class of flavin-dependent halogenases, which we've been talking about. And again, as he mentioned, these were enzymes that were capable of halogenating at these really electron-rich substrates. So these aromatic rings, like you see in the case of ribacomycin. But if you recall back to the beginning of the talk, I showed you this diversity of structures of molecules that can be halogenated, and not all of them are halogenated at these electron-rich sites. And so this led to this question, what about halogenation of an sp3 carbon, an aliphatic carbon? And so a great example of this is seen in this molecule here, syringamycin E. This is a depsipeptide isolated from a bacterial strain that's a known plant pathogen. And this molecule is a key virulence factor during infection of the plant. And it's a non-ribosomal peptide, but for the purposes of our talk and what we're interested in is this residue here at the bottom of the slide, and you can see it's a chlorinated threonine residue. And this halogenation is very different than the halogenation we're seeing from these flavin-dependent halogenases. This is an unactivated carbon. And so then when you look at the gene cluster that's responsible for the production of syringomycin E, there's also no flavoprotein homolog present in the gene cluster. And so this kind of makes sense, right? We're seeing a different type of chemistry here. We might expect a different enzyme is performing this reaction. And so Chris noted the presence of this non-heme iron alpha-ketoglutarate dependent enzyme instead. And at the time, these were only recognized as oxygenases. They weren't known to perform any kind of halogenating chemistry. These enzymes are really difficult to work with, and so Fred is going to tell you a little bit about some of the challenges that are involved in actually purifying and working with this really difficult class of enzymes.
Yeah, so as Chris was gearing up to start working on this class of halogenase, prior to joining the lab, we were brainstorming as to how it could be done and things like that. I was like, you probably need, a glove box, right? And bring this back to your lab, which I didn't know at the time. I didn't say that. I was like, you need a glove box. I was like, oh yeah, I had one back at MIT in the mercury reductase days. So it was like, yeah, that makes total sense, right? So then, kind of build the instrument park and got, an actor to purify the enzyme linked to a glove box, and that really what enabled this whole effort. That was a lot of fun. And then radioactivity, as I just said, right, played a key role there. And fun fact on the radioactive chloride, turns out that it never goes away. The half-life is 300,000 years. So it's basically radioactive, and you measure it on day one, you measure it 2 years later, it's exactly the same count that you'll get, very unlike the other ions. For those of you that are familiar with radioactivity, is less and less common. And energy-wise, it's like Peter D3, so you shield yourself behind a plexiglass and you're good to go. So it's not that bad. But it really never goes away, so it's not expensive to purchase radioactive table salt. It's expensive to get rid of it. As we were, building the data sets for these papers, we were getting, waste generated, by the book and everything, stored in barrels and that sort of thing. At some point, we're like, well, we're getting close to a barrel. We should figure out how to get rid of it because it's not going away. The lab is not going to be around for millions of years. So talk to the radio safety office of Harvard Med School and everything, and get a quote and things like that. And turns out getting rid of that barrel was in the well above $30,000. So it's probably the most expensive garbage bag that went out of non-clinical research at Harvard for quite some time, I suspect. But nevertheless, it could be done, and that's really what helped pioneer several of those papers, having identified those radioactive intermediate.
All right. So that's a lot of work just to purify our enzyme of interest, SIRB2. And I think, to me, this is one of the best parts about this story, is when you look at the paper, you can see this key finding that, yes, SIRB2 is a halogenase. But what you don't necessarily read about and what we get to hear about today is what it really took to uncover that discovery, these really technical challenges that the lab had to overcome. And so what I'm showing to you here is this SIRB2 reaction. And we can see that it is capable of chlorinating threonine. So on the right side of the slide, these are HPLC traces. And we can see with and without SIRB2. When SIRB2 is present, we see the formation of our chlorinated product, chlorinated threonine. And we know this by comparing to standards at the bottom trace here. So I think that while we know that this is a halogenase that acts on threonine, that kind of barely scratches the surface of what it really took to come to that conclusion. And so they were also able to, the Walsh lab was also able to show that SIRB2 required iron and alpha-ketoglutarate as co-substrates. And this was important because it demonstrated for the first time that this class of enzymes, this non-heme iron alpha-ketoglutarate dependent class of enzymes, were responsible for halogenating an aliphatic carbon. So we can see it does require these co-substrates of iron, alpha-ketoglutarate, and it was also oxygen dependent. So actually, this reaction is even more challenging than I've let on. Generating the substrate for this assay is also complicated. So as I've shown on the slides, but have kind of conveniently skipped over talking about, our substrate for SIRB2 is actually carrier protein bound. This is a assembly line system, which you're going to hear about a little bit later today. But that means that SIRB2 actually would only chlorinate the amino acid threonine when it was tethered to a carrier protein. It actually wouldn't chlorinate on the free amino acid. And so this is non-trivial work just to generate the substrate for our enzyme of interest, SIRB2. And so I think that a lot of the beauty of this work really lies in the experimental and technical details that went into the project. So as I mentioned, SIRB2 is oxygen labile, and so that's why it necessitated that glove box setup that Fred showed to you all. And that picture actually is of his glove box in Chris's lab, which is pretty neat. Second, I explained to you all that our substrate is carrier protein bound, right? And so because of this, the product detection actually requires the addition of another enzyme, a thioesterase, which is capable of cleaving off our chlorinated product so that we can detect it via HPLC or mass spec. So that's another enzyme in the mix that we have to add in there. And then again, because stemming from the fact that our substrate is carrier protein bound, that means the amount of product that's coming out of the reaction is stoichiometric with the amount of SIRB2, our carrier protein, that enters the reaction. So we're talking about really small amounts of product. And so in order to characterize this, Chris and the Walsh lab had this really lovely technique of derivatizing the amino acid so that we could see this fluoroisylindol derivative that really facilitated detection. And so it's this combination of these really creative solutions, I think, that allowed Chris to determine that it was, in fact, this non-heme iron halogenase that's capable of generating an oxoiron reagent that's able to halogenate what we call an unactivated carbon. So this is a reaction that's really challenging to do in synthetic chemistry. And being able to do it with the precision of this enzyme is kind of the holy grail of synthetic chemistry. And so being able to characterize that for the first time was a huge achievement and came with a fair amount of challenges as well. And so a lot of questions, of course, remained about the mechanism that SIRB2 actually uses to do this halogenation reaction. And so in collaboration with the Drennan lab, they were able to obtain a crystal structure of SIRB2, which is what I'm showing to you here. And if you look at the active site of the enzyme on the right side of the screen, we see our iron in brown, and then the chloride present in green. And what's interesting to note about the active site of this enzyme is you can see these two histidine residues that are coordinating the iron. These are really important and were known from the non-heme iron alpha-ketoglutarate-dependent oxygenases that I mentioned to you earlier. But in the oxygenases, there's an additional carboxylate ligand that's present in the active site. And they didn't see that in the active site of the halogenase SIRB2. And so they were actually able to take advantage of the promiscuous halogenating activity of SIRB2 that it could also brominate. And by looking at a structure with bromide, which has a very distinctive diffraction pattern that you can see in crystallography experiments, they were able to unequivocally demonstrate this iron halide bond, which was seen for the first time. And we now know that from the structure we can see that the halide takes the place of where the carboxylate ligand is in the oxygenases. Okay, and so that crystal structure led to a working mechanism for how SIRB2 actually operates. And we don't need to talk about the whole thing, but I think the most important part to note is this high-valent iron 4 species at the bottom. This is really the key intermediate of this reaction. And it's the decarboxylation of alpha-ketoglutarate that generates this high-valent oxoiron species that's capable of abstracting a hydrogen from our substrate, which can then, by radical rebound, we see chlorination of our substrate and then regeneration of the iron center. And so it's really radical chemistry that is driving this ability to halogenate at an unactivated carbon. And so that's how nature gets around this really complicated chemical reaction that's really difficult to perform in a lab. And so just to give you a sense of the type of work that this discovery enabled, by understanding SIRB2 and this new class of enzymes of non-heme iron alpha-ketoglutarate-dependent halogenases, Chris was able to look at this molecule. It's a really beautiful structure, chronotine, also produced by a bacterial plant pathogen, a Pseudomonas species. This structure mimics a plant pathogen, methyl jasmonate, and so during plant infections, it actually can make the plant more susceptible to infections. And Chris was able to look at this structure, and you note the presence of this. We've seen this molecule before, and we've talked about this really unusual three-membered ring, the cyclopropyl amino acid there. And by looking at the gene cluster, they were able to see the presence of a non-heme iron alpha-ketoglutarate-dependent enzyme that looked like SIRB2. And they were ultimately able to show that it's chlorination of this carrier protein tethered amino acid that leads to the formation of this three-membered ring. So we have halogenation by this enzyme CMAB, followed by an intramolecular elimination of that chlorine to form our cyclopropyl amino acid. And so with that, I'm going to pass it over to Fred to tell you a little bit more about the legacy that this work leaves. But I just want to say that I never had the chance to meet Chris, but his work impacted so much of mine. I used techniques from his lab during my time in graduate school, and so it's been a real honor to get to share some of his work here today. Fred.
Thanks a lot, Kate. So there's a lot of words on this slide, but it's really to do justice to Chris. How did he really make all of this happen, right? Think of it. It's a single atom change on like a 60 kilodalton protein that is post-translationally modified. But imagine yourself as a young faculty tackling such problems. It's like impossible, right? But Chris made it happen because, first, he was a visionary and could see how it could be done, right? All the tools were now available in his lab, right? Purified SFP was now common to put those PPAN group on the machinery. Recently back then, right, the thioesterase, tool as a thioesterase to cleave off those amino acids had just been identified, and then we could, cleave off those amino acids and characterize the derivatives on HPLC. But these are non-aromatic amino acids, so we needed to use tools from the amino acid analytical work and derive them to kind of start seeing them in HPLC traces with sharp peaks. He had an impressive instrument park, right? All the basics were there, LCMS, you could do radioactive HPLC, it's common, but not that common, and everything was there. But on top of it, the people, right? The people. Chris had an eye to hire complementary expertise and people that were highly creative, and a lot of that team were hardcore synthetic chemists by training that wanted to learn chemical biology, right? So, and I'm more an enzymologist by training that went to Chris Lab, so I can make molecule, but by no means I'm not very talented in the hood. So, but by teaming up with synthetic chemists, we're like, planning ahead projects and things like that. Oh, we'll need these standards, and these standards, and within a few weeks, you had all the tools available to characterize your system, right? That's unheard of. Like, not many labs can do things like that. So it's really all about the people. He really pioneered the work, and then teams up with collaborators from other fields to push it further, and then it gets pushed even further by others, as you'll see on the coming slides. So Chris's inspiring work on halogenases had an impact on at least 4 fields that I could identify. First, there were several crystal structure and a deep mechanistic understanding that followed. Chris was involved in both of those aspects. Then some other fields also were really inspired by this. There's bioinspired catalysis that came to play, and recently these enzymes started to be engineered more efficaciously. So then it's going to be a lot of animation with various papers on the different categories, right? So as Kate mentioned, Chris teamed up with Cathy Drennan's lab at MIT to figure out the SIRB2 crystal structure. And fun story, we were basically, okay, we want a structure of this, and we were looking for a crystallography lab that had a glove box. It's a pretty niche market, right? We kind of found a few names, and then there was this emerging faculty at the time, Cathy Drennan, that had built an amazing website, and you could see it, she was publishing on metal enzyme, and you could see her glove box. Well, so she rose to the top, former department of Chris. It's like, I'm just going to write to her. And, she's probably starting to laugh. She's following online, I'm sure. At first, when she received the email, she thought her email had got hacked by some of her grad students and that they were playing a prank at her. So she made them promise that it wasn't her. So then she did reply to Chris, and of course she wanted to embark on the collaboration. And then, the rest is history. They had quite a long collaboration together. And given that MIT, Harvard Med are pretty close, within a few days, we were in Chris's car, meeting face to face and planning this whole thing, right? Same when the crystal came out. So it was really engaged and there and following the science on a daily basis. They had several papers together, and since then, several other groups have contributed quite a lot of knowledge to the field as well. So it really, his work pioneered the whole thing. And then when it comes for a deep mechanistic understanding, Chris also had a significant impact, right? That's the chloramine intermediate, that potential that was uncovered in that paper. Like it was pushed further by Chris in collaboration with a former postdoc of his, actually Marty Bollinger, right? He's a double-trained stubby PhD, Walsh postdoc now at Penn State with Carsten Schreib, right? They really pushed that field quite far, and even teamed up with someone right here, Ed Solomon, in the chemistry department, that has quite a vision with very exotic spectroscopic technique, could really push this to an electronic understanding of the whole system. So it's quite impressive work. I was able to, I have many quotes from all these other groups, but I was able to dig two, like word to word, right? This is from Marty at Penn State. This research influenced the direction of several academic research groups, including my own, for the next decade, right? They worked, from Chris's inspiring work for at least 10 years after that. In terms of bio-inspired catalysis, that's, a field we don't hear a lot about necessarily, but there's quite a lot of people active in that field as well. I remember attending the International Conference for Bioinorganic Chemistry in 2005 to present some of that work, and the halogenase for the bio-inspired field was really the buzz of the conference back then. It's called HICBIC. It's still, that conference still exists in that field. And it took them years to even come close to nature in synthetic catalysts, but they got there, and Larry Kay at U Minnesota really took that problem at heart and really pushed it to the limit. And here is a quote that I found from him from a few years back that really shows how this work really inspired this whole field of bio-inspired catalysis. And then more recently in directed evolution and engineering, it's not an easy enzyme class to change substrate preference and everything given the mechanism at play, but some groups are really getting there at succeeding at evolving this whole class to do some very interesting chemistry. So biocatalysis and directed evolution are benefiting from this as well. So we can see it really inspired quite a lot of work, and as we're preparing this presentation, it's like, how can we wrap this up, right? We don't want to do a classic summary of the science, but more about how did this really contribute. So we had the idea to go in, when someone publishes close to 1,000 articles, you can start looking at what's emerging of a top 100. You can't say that about a lot of people, but we can for Chris. And it turns out that in his top 100, there are nine articles that are of the most cited articles are from that field. 2 from the flavin halogenase, 4 for the non-heme, and 3 of his amazing reviews, right, really emerged to the top and really shows the dramatic contribution that Chris has had on this field. So with that, just want to thank everyone involved in the early and mid days of the halogenation part of Chris's career. This is by no means everyone, right? A lot of people work on halogenases following that time as well, and several collaborators were involved. This was the early days of digital camera, so I was able to dig out a highly pixelated picture, but that really shows, how Chris was the large group that it's those joint conferences with David, Chetan, and Mohammed that was joined. This is actually at Wellesley College where Diana hosted that conference that was being held, I think, at Babson probably at the time. So with that, thanks a lot, everyone, but more importantly, thank you, Chris, right, for all his contribution to the field.
Should we take questions?
Yeah. Questions? I'll say, while we're queuing up the first question, that I remember distinctly the moment when that crystal structure was solved and he had seen it for the first time. That was probably, there was more visceral joy in Chris on that day than I had ever seen at any other time. He was running around with childlike enthusiasm, wanting to show everybody the structure of the iron bound to chlorine or bromine. So just a true moment that was emblematic of something he'd been wanting to figure out for so long, and just knowing by seeing that this is how it worked, I'm sure he would have said that was one of the moments in his research career he would always remember. Quick questions for Fred and Kate.
Hi, great talk. Thank you, guys. I'm curious, when you were talking about engineering and evolving other halogenases, is that like site-specific halogenation? Is it substrate specificity? Is it specific to different types of carriers, depending on what amino acid precedes the threonine? And like, yeah, what type of work is going on in that space?
Yes, I'm following that space, but not that closely either. But I think the main goal is changing specificity, right? Most of those enzymes really do the exquisite reaction that they evolved for in nature. And I think the academic groups that are tackling that problem are really trying to make those enzymes more general catalysts to really modify various natural products and potentially lead to even more bioactivity, or use them as a biocatalyst to do like enantiomeric kind of intermediates, right, instead of being racemic, that sort of thing. So that's usually where it's applied.
I just wanted to, and this is Ellen, so that was my PhD work, and I haven't thought about it in a very long time. However, I did want to just comment, I think when Michael was saying about Chris running around the lab excited about data, I do remember becoming quite cheeky by the time I was a senior grad student, and that data that came in that showed the long-lived intermediate, I didn't tell Chris. I just posted a TLC image with some labels on it on his door, and he came back from some long trip. And at some point, I was pipetting it, and I saw him running down the hall towards me, and I still remember the excitement and light in his eyes. And I think that's something that I won't ever forget about Chris. Yeah.
Maybe time for one last question. If anybody's got, oh, I see a hand up right here.
Could you talk a little bit about where the organisms that make these live? Are they in high salt concentrations that they just need to do something with the chlorines, or are they like evolving and specialized in a niche that's kind of more general?
Yeah, so these bacteria they don't live in a high salt environment, right? The Syringomycin one and the coronatine one, right? Both of these natural products are plant toxins, actually. The coronamic acid is actually can kill tomato plants. We had pictures of that. And it's basically contributing to bioactivity, right? And there's enough salt in the cytoplasm that it can get done, but they're not from Yellowstone or that sort of source.
Although I will say there are a lot of halogenated natural products found from marine organisms, like marine bacteria.
Yeah, so that's definitely true. In the ocean, where obviously, yeah. But not these particular ones.
Let's give Fred and Kate a round of applause. A final round of applause. Okay, next up is our second tag team pair, and they're going to talk about the logic of assembly line enzyme function in making these massive natural products that Chris was obsessed with. The first of our pair is Rahul Kohli, who's a professor at Penn and an infectious disease doctor. And Rahul is generally distinguished by being an all-around delightful person. And also, when I asked him only moments ago what years he was in Chris's lab since he left just before I arrived, he told me 2000 to 2002. And I did the math in my head. Some of you are doing this now yourselves, especially if you're a trainee and you're thinking, Michael must have gotten that wrong. I did not get that wrong. The other distinguishing thing about Rahul is that he did what is quite possibly the shortest PhD in Chris's lab or anybody else's lab at just over 2 years. And consider that when you think back to how long it took you or is taking you to make it through your PhD. And he is joined by Alex Suhu, who is a current trainee in Chaitin's lab, and in a way that's entirely fitting the topic of their presentation, is working on fundamental aspects of the logic by which these massive enzymes work. And so they're going to educate us now.
Thanks, Michael, for that embarrassing bit of information. It tells you that Chris wanted to get rid of me quickly, most likely. But, hi, everybody. I'm Rahul Kohli.
Hi, I'm Alex. I'm a graduate student in Chetan's lab in chemical engineering.
So we're going to be tag teaming this session as well. And our goal here in this, this is an inspired setting, this idea of a teach-in. And there's a lot of things here that we hope to sort of convey in teaching. Some of it is about the science and the way that Chris helped to shape these many different fields. Some of it is teaching about who Chris was and getting that picture of who he was. I think hearing that moment from Ellen about Chris running down the hallway with that enthusiasm, there's a lesson in there for all of us. So I guess to start the teaching, I'll start with my foray into the Walsh lab. I was an undergraduate at the University of Michigan, and I worked with Vince Massey, a flavor protein enzymologist, for my 4 years as an undergrad there. And when I left Michigan and headed up to Harvard for my MD PhD training, Vince said to me, in a very serious and stern tone, there's only one place for you to do your PhD, and that's with Chris Walsh. So there I was, right, an early med student looking for my lab, and I emailed Chris hoping for the opportunity to work in the lab, and I didn't get a response. So I emailed again, and I still didn't get a response. So my third email, I got a response, and I got to sit down with Chris finally. And one of the first things he said to me was, it's a good thing that you email me more than once, because I like to know who's serious before I talk to them. So lesson in there for all of you, to persist, to not miss out on, on life-changing opportunities. Sometimes it takes more than one email to get there. So we're going to be focused here on the question of natural product assembly lines. And today you've seen a lot of diagrams that have gone up with these huge enzymes that have been captured as a bunch of strung together boxes. And so one of the objectives of this teaching is to bring some light to those boxes. And that's what Alex and I are gonna hope to do. So I think the place to start here is to just envision this from Chris's eyes. I think Emily Valskis did a great job this morning of showing you that image of an inspiring piece of art. And to Chris, looking at molecules like this, if you looked at them through his eyes, and hopefully many of you will appreciate the same way, these are spectacularly beautiful molecules that nature makes. Just take a moment to stare at what's in there. And I think in the session that we just had, you could take some piece of it and say, this particular atom that gets added is spectacularly beautiful and complex. How did that come to be at that location? And we heard about the fascinating chemistry that can dictate these individual sort of changes. There's another aspect to what is this spectacular diversity and this beauty in these molecules, which is that in that complexity, there's also a simplicity and a logic. And so we'll hear more about antibiotics later today, which were one thing that Chris made major contributions to. In these molecules here, you see molecules that were inspiring for their medical interests, but made and isolated from various natural sources, including Vancoymcin, which we're sure to hear about later today. These same sort of lessons about beauty in the molecules that nature makes extend over to other classes of molecules as well, like these anti-cancer agents and immunosuppressants that are there. But coming back to this idea that in these molecules there is both spectacular complexity in the small pieces, but also logic and simplicity. These questions of how a molecule like that can be broken down into similar and smaller components is one that was a major focus of the Walsh lab for many years, and is going to be the topic of our discussion here. So when you take a look at a molecule like vancomycin, you can see that as you take that step back and look at what's there and what's complex, built into that into the molecule is that simple linkage of a peptidic linkage, simple amide bonds that are being formed with building blocks that are going into making that. At the level of that of the genetic information that encodes for the natural generation of these products, one can see gene clusters, and one of the nice things that nature did for us is to put many of these assembly lines into a cluster so we could start to look at and understand what that specific cluster does. But in the case of vancomycin, where we can trace that backbone to 7 monomers and to 6 peptidic linkages, you can see core proteins that are involved in the biosynthesis of these molecules that themselves have a structure that makes sense and brings out that simplicity, where those 7 different building blocks each appear to have what is a module dedicated to each of them, and in the process that could encode for their generation. And so, again, one of our objectives in this section of the teach-in is going to be to take this and to say, well, if these modules are each responsible for one building block, and then the whole molecule needs to be pieced together, how do we build up towards piecing molecules like this together? And we're going to do that as a group as we walk through the science that Chris's lab helped to shape. When you see this idea of this kind of modular approach to these modules, it brings out immediately the idea of something of an assembly line that can be used to construct that final elegant product that's there at the end. So in this particular situation, this idea of assembly lines had precedent in different areas of biology as well. One of the ideas of sort of building up things from simple precursors came from fatty acid biosynthesis, where modular building blocks are used in an iterative fashion to keep adding 2 carbon units to fatty acids in the process of their biosynthesis. This example is one that was important and really helped to shape thoughts about this process more generally. At the heart of that building block process of fatty acid biosynthesis is the idea that the intermediates that are being built up during the assembly line need to stay on the assembly line, on that conveyor belt that's moving across the different points of synthesis. And the way that those intermediates could stay tethered to the building block and move along the assembly line was through an extended arm from a phosphopantethene group that's derived from the molecule CoA. So this sort of makes a starting point for entry into thinking about natural product biosynthesis. And Alex is going to take us through thinking about how it is that this fundamental piece that's essential for the assembly line, that arm, is going to get built up, and hopefully give you some, in the process of telling that story, some picture of what Chris was like in the process of that discovery. So Alex.
Thank you. All right. So Chris decided to jump into the field of natural products due to his interest in metals, actually. And so he was really interested in enterobactin, as you heard earlier, due to its role as in iron sequestration. And so it was well known that enterobactin was encoded by the gene ENTF, and it produced a non-ribosomal peptide synthetase. And what Chris soon realized when he jumped into this field was that while he was able to make NTF protein, he was unable to get high quantities of active protein. And what he realized was that the issue was that the NTF he was purifying didn't have the covalent pantethene onto it. What he was purifying was its apo form rather than its active hollow form. And so Chris's main question became, what is the enzyme that catalyzes the conversion from the apo inactive protein to the hollow active protein? And then you can see it's drawn as a squiggle, which is indicative of this phosphopantethene arm, which looks a lot like CoA. And so you can imagine this is the early 1990s. How do you go about finding an enzyme when you have no idea what its sequence is? All you have an idea of is what its function is. And so they decided to start by looking for the ACP synthase because they knew that enzyme had to go and exist due to its role in fatty acid biosynthesis. And so you can imagine you don't know how the enzyme is produced, how much is being made, so you need to get massive quantities of E. coli. And so I read in his paper they bought literal bricks of E. coli paste, which they then homogenized in the French press, and then they ran it through a column to fractionate it and try and make it easier to work with and limit the number of candidate proteins in each fraction. And you can see that going from the lysate through an anion exchange column or a cation exchange column, it cleans up the sample a fair bit, but it still has way too many candidate proteins to figure out what the actual ACP synthase is. And so they needed some assay to monitor activity, and so they decided to use a radiolabel activity assay. They had ApoACP, they added radiolabel CoA, and then incubated it with different fractions they had from these different columns. And then they monitored how many of these ApoACP samples had activity via scintillation counting. And so while they're able to get active fractions, they weren't able to get highly active fractions. And so they needed some way to go and actually enrich for the amount of ACP synthase in their samples. And so they decided to take an activity-guided approach. So they went and conjugated the ApoACP onto some resin and then flowed crude lysate through it. they knew that the ACP synthase had to have some affinity for an ACP, and this ACP synthase would go and interact with the resin-bound ACP, while all the other proteins would go and just flow away. And they did a good job at making the sample substantially cleaner. And you could wonder, oh, is the ACP synthase that band between 31 and 45 kilodaltons? It turns out that previous studies suggested that it was actually a 50 kilodalton protein. And so they cleaned it up a bit more, and in their final sample, they had some highly active fractions. But what you see is that even though they had highly active fractions, they didn't see anything in the Kamasi gel. And so the question became, where's the band? And to go and tell a little bit about the story, we have Ralph Lambalot, who had a little recording for us, and he was the lead author on this study.
So it had been 27 years since Roy Vagelos' lab had published on the purification of what they had dubbed the ACP synthetase from E. coli. And in that paper, they reported a molecular weight of about 50 kilodalton by size exclusion and sucrose sedimentation. So I was now a year into my postdoc with nothing to show but some very highly active fractions of ACP synthase activity and a stack of dry-mounted silver-stained gels that clearly showed nothing in the range of even 25 to 50 and beyond kilodalton molecular weight range that had been co-purified with activity. Then one morning, Chris walked into the lab, leaned against my bench with his arms crossed in front of him, and he said, I was traveling, and I had a dream. And in that dream, ACP synthase was of remarkably low molecular weight. Put his hands up. He said, do with that what you will. Turned around and walked out of the lab.
And so I think everyone who has heard that story realized it was a very classic Chris story. And Ralph decided to take Chris's advice. And so he pulled down some of his old gels, and at the very bottom, near the buffer front, he saw a very thick smear, and he always attributed it to proteolytic digest or something along those lines. But given the sage advice from Chris's dream state, he decided to go and rerun his samples on a different type of gel, which was better at resolving low molecular weight proteins. And sure enough, he ran it, and there at 14 kilodaltons was the ACP synthase. And so this was great. He cut out the band, he ran over the core facility, and within a day, they had the sequence of ACP synthase. And so this was fantastic. They finally had an enzyme that was able to catalyze PPAN transfer. But this shifted the question now back to enterobactin synthase. You have this inactive ENT-F, and you know there's an enzyme that catalyzes it and catalyzes the PPAN transfer and generating active protein. Is this enzyme ACP synthase, or is it a different related enzyme? And so they used ACP synthase in their homology search, and they found 3 candidate proteins, O195, ENT-D, and SFP. And they repeated the same radiolabeling assay, and they had ApoACP, and they incubated with ACP synthase. And as expected, they saw that it was able to catalyze PPAN transfer. They then went and excised the PCP domain from the ENT-F, enterobactin synthase, and when they incubated with ENT-D, they also saw that it was able to catalyze PPAN transfer. And so finally, Chris and coworkers had found the elusive ENT-F PPAN transferase. And this enabled study of the role and biological activity of ENT-F. And so this was fantastic, and what they also realized was that while ACP synthase was very good at catalyzing PPAN transfer with ApoACP, you could see that it didn't have much activity at all with the ApoPCP. Likewise, ENT-D had great activity with the ApoPCP, but it didn't have much activity with the ApoACP. And so what they concluded was that ACP synthase and ENT-D are specific PPAN transferases. And so this was really great. Chris was able to overcome the bottleneck in studying enterobactin biosynthesis. But there's many valuable assembly line natural products, and now the question shifted to how do you go and study these molecules? What enzymes are going to charge these assembly lines? Is he going to have to find a ppm transferase for each of these cases, or is there a generalizable solution? And what you'll probably notice from this other slide is one candidate protein, SFP, which seemed to work in both cases. And so Chris and coworkers did some more deeper understanding of SFP, and they tested out its activity with various carrier proteins. And they saw that it worked well with different NRPS modules from a surfactant NRPS. It worked well with other enterobactin biosynthetic genes, and it also works well with lysine and fatty acid synthase enzymes as well. And so what they concluded was that SFP was compatible with many different carrier proteins. And this was extremely valuable because a lot of these natural products come from very hard to work with organisms. And so approximately 3 years or so later, a strain known as BAP1 was generated, and that enabled the study of these natural product assembly lines through transferring the genes from these host organisms, putting it into E. coli, and while normally the apo form of the protein would be produced, by integrating SFP into the genome, you could just take the genes, add it to E. coli, and you get active hollow protein. And this really opened up the door to study many different assembly lines and gain insight into how they actually work, which is what Rahul will talk about.
Thanks, Alex. All right, so now we have a way to charge assembly lines, and we're going to come back to the molecules of interest and try to build our way up to understanding the entire process, the simple logic that exists in making these complex natural products. So to return here to a molecule like vancomycin, and to the core of what's so interesting about the process of these non-ribosomal peptide synthetase machineries, some of the building blocks that go into it are things that we can readily recognize. So in the example shown here, natural asparagine and leucine are building blocks that as you start to retrosynthetically dissect this molecule, you can readily recognize. But importantly, one of the strengths of these approaches that nature takes in this biosynthetic pathway is that the building blocks are not limited to those 20 canonical amino acid building blocks. There's a scope of hundreds of building blocks that can, in fact, go into the assembly line, offering the ability to then exponentially increase the diversity of the resulting natural products. So as we've seen in some of the elegant chemistry that goes earlier today, each of these building blocks then themselves offers an opportunity to explore new and interesting chemistry. And in this particular case, you can see different phenylglycine building blocks with dihydroxyphenylglycine as an example of what goes into the complex natural product of vancomycin. So diversity of building blocks is our starting point, and now each of these building blocks has to get incorporated. For thinking about how these building blocks get incorporated, we can take a molecule, a natural product, and start to dissect it further. So here's an example of a cyclic decapeptide, gramacydin S. When we break down the structure and start to, again, retrosynthetically break it down, we see 10 amino acids that go into the construction of this that are linked together by 10 total peptide bonds because of the cyclic nature of this molecule. So how do these individual building blocks get built together in that assembly line structure? So analogous to thinking about ribosomal synthesis, there must be mechanisms at play to take a simple amino acid and make it so that it's ready to be incorporated. And this then brings us to the function of one of the boxes that are of interest, those A domains, or adenylation domains. So shown here is the gramacydin S A domain, the first domain, which takes as its substrate, canonical substrate, phenylalanine. But distinctively from ribosomal synthesis, there can be some degree of promiscuity in these domains. And so in this specific instance, the adenylation domain of gramacydin can in fact take other amino acid building blocks into the initial product, which yields then natural products that might be somewhat diversified at that position. So some aspect of distinction, but there also needs to be selectivity in nature, and we're going to see how that selectivity can get enforced at next stages of synthesis. So here, the adenylation domain catalyzes the simple chemistry analogous to the tRNA synthetases of activating the acid portion of the phenylalanine, followed by its conjugation then to the phosphopanathene arm that we just learned got appended by a phosphopanathenol transferase onto the carrier protein domain. So the joint action of that A and PCP domain leave you with a loaded domain in this particular setting. Here, in this specific instance, this is followed by an epimerization domain, a domain that can change the stereochemistry of that building block. And that, as we'll see in a moment, can be a factor that influences selectivity in downstream steps. So now having built, loaded a specific module with a specific amino acid, how do they get built together? How does the assembly line logic continue? So in other work identified once again here from the Walsh lab, we have the conjugation chemistry that's relevant to these processes. So once you've loaded neighboring domains with specific amino acids, the act of linking them together is the function of the C, or condensation domain. So the condensation domain is going to catalyze that simple peptide bond formation. But again, thinking about how nature is different in different circumstances, from that ribosomal chemistry where the peptide bond is always going to be the same at two neighboring sites, here each of these condensation domains is catalyzing a unique type of peptidic bond linkage. So in this particular case, a linkage between module 4 and 5 of tyrosine, a cyclic decapeptide antibiotic that's somewhat like gramicidin. This molecule here involves the conjugation of a tetrapeptide from this intermediate state to the 5th unit building block extender. And that conjugation is going to be what then moves the molecule down the assembly line to after the formation of the next of that peptidic bond. So in these examples here, that condensation domain can serve as something of a gatekeeper, making sure the right kind of peptide bond is formed and potentially exerting selectivity. We'll see in a moment how loading these domains with different kinds of peptides can actually teach you about specificity in these systems. So you're looking for a logic of simplicity and of specificity in these systems. In this particular case, once again, we have this epimerization domain, and the stereochemistry at that position dictates whether or not molecules move forward through the assembly line or whether intermediates that might not be charged in the right way are cleaved off at an early state. So we built up now this idea of loading the assembly line with individual monomers, putting them together into longer molecules. But at the end of the assembly line, there also has to be a way to unload the cargo. So here's the example of the final domain in in tyrosine synthesis. That final domain is going to have a fully charged decapeptide on that arm, and it has to be unloaded. It's built as on a thioester tether, and so that unloading function is going to be catalyzed by a thioesterase domain, or a TE domain. The TE domain contains an active site serine that is going to attack and make the ester intermediate, picking up from the thioester and moving downhill in the assembly line. And this then can catalyze release from the system, or in the case of much of the chemistry that we've seen, it can catalyze other kinds of release reactions. So an example shown here is one of a cyclizing thioesterase domain, where the N-terminal end of the peptide cyclizes back onto the end of the molecule to generate this end cyclic peptide product. So studying these sort of domains in simplicity are the kind of approaches that can allow for additional flexibility in these systems. So shown here is the idea that if you want to study the biochemistry of this system, you may not want to study it in the full context of the assembly line. And so starting to think again about the phosphopanathene arm that we discussed early on, are there ways to chemically start to mimic parts of these assembly lines? And so what Alex and I want to end on in this section of the teach-in is how chemistry gets brought together with biochemistry in these systems. And the chemistry can take place in thinking about these intermediates. So shown here is an example of where in lieu of the decapeptide loaded onto that carrier protein that was the product of 10 different modules bringing together the molecule, one can make a decapeptide that's simply activated as a simple N-acetylcysteamine thioester, an SNAC. And this is meant to mimic the end of that phosphopanathene arm. By using that surrogate of the natural substrate, you can study it with the thioesterase domain and catalyze the generation of those cyclic peptides. So continually looking for simplicity and bringing chemistry to bear on these enzymatic questions was another consistent lesson of what came out of Walsh Lab's work. So we've built up all these intermediates, and now we just want to, so again, a goal here is to understand and to learn the simplicity of the logic that Chris taught us about. So with this all built together, can we come back to that simple example of enterobactin that brought him his initial interest into this field, and think about how a molecule like enterobactin is made? So many of you maybe now could take out a piece of paper and start to sketch together a retrosynthetic approach to a molecule like enterobactin, but I'm not going to make you get out graph paper and do it like Chris might have. We'll do it together to think about how this molecule is made. So starting from building block units shown at the top. These building block units can be constructed and then loaded, activated by Ent-E, and loaded onto the Ent-B module with a squiggly arm with the phosphopanathene on the end of it. We have a loaded monomer now with the dihydroxybenzoate building block built onto it after the adenylation step. In the second module, we have an A domain that takes serine as its substrate and loads it onto that PCP domain. We've learned about what the condensation domain does. After the condensation domain's action, it's going to put these two things together and make this smaller building block linking together the dihydroxybenzoate with that serine. The thioesterase domain, which can now unload, can also do more than that. It can unload once, go back, pick up another intermediate to dimerize, go back again, trimerize, and finally catalyze cyclization to bring us to that final enterobactin molecule. So hopefully in that you've seen the simplicity of the logic that's there, and through consistent studying, breaking it down to individual components, a molecule like this, that again is just spectacularly beautiful in its function of iron chelation, can be built up from the logic of the work that came from Chris's lab and from others, collaborators through time. So with that, I'm going to hand it back over to Alex to take us to the next step of chemical biology here.
Thank you. And so Chris taught us a lot about NRPSs, but they're not the only assembly lines out there. There's also polyketide synthases. And so polyketide synthases, if just through the schematic you notice, it looks a lot like NRPSs. So you notice that it's also made of many modules, and each module has a repetitive set of domains. And in this case, each module is responsible for a ketide extension, so a 2-carbon chain extension. And so as the propanol unit passes through the polyketide synthase, it'll eventually go and become the 6-deoxyethronolide B, the aglycone precursor to the antibiotic erythromycin A. And you'll notice that polyketides are also able to generate a tremendous amount of chemical diversity. And so fortunately for us, nature has found a way to bring together NRPSs and PKSs in hybrid NRPS-PKSs. And so this is an example, apotholone, and this is generated by a hybrid enzyme, and you can see the keto groups come from the PKS and the N indicating the amide bond formed by the NRPS. And so anyone who's studied assembly lines has probably been enamored by the idea of engineering and tinkering with these systems. Can you go and change a module and swap it out with a module from another assembly line and generate an analog of 6-EB or any of these other assembly lines? And so I think Chris referred to them as unnatural natural products. And so you can imagine doing that, and unfortunately, you're not likely to be very successful because these enzymes have built-in specificity and selectivity. And so let's say you want to go and study a standard assembly line. In this case, you'll have a bi-module in an NRPS system. So typically, you'll find NRPSs come on very long proteins, which contain multiple modules which are covalently linked. And so you can imagine if you ask the question, how does the acceptor residues amino acyl group affect the condensation rate? It can be very difficult to study in the native setting. Fortunately, we're able to jump into the middle of the assembly line using some enzymatic and chemical tricks. And so while we were unfortunate in the case of enterobactin synthesis, when you were able to only produce APL, Ent-F, when you didn't have SFP, this was actually a pro. So in this case, if you purify just module 2, you're going to produce its APL form. And then if you provide SFP and an amino acyl CoA, it will go and charge the module, but also throw in an amino acyl group onto the module. And so then you can ask the question, how, if you go and use different amino acyl CoAs, how does that affect the rate of condensation? And so Chris was able to go and study this and understand how putting these assembly lines into unnatural situations affected its ability to function. And so this was a nice example of jumping into the middle of an assembly line for a mechanistic dissection of how NRPSs work. And so SFP, it turns out, while being promiscuous for which carrier protein it can work with, it was also promiscuous in which substrates it works with. And so in the classical example, it will go and take ACP in its apo form and convert it to its holo form using CoA. But you could also use something like benzyl-CoA. And it's going to not only charge the ACP, but also throw on a benzyl group at the end of it. And Chris and coworkers found out that SFP was compatible with a lot of different substrates. And so SFP has proven to be an extremely valuable tool for studying these assembly lines in different contexts. And so you can also imagine, as you saw with Rahul, that you could go and skip the assembly line altogether using an SNAC substrate. And so this kind of really inspired Chris and coworkers to think about what else does chemistry have to offer that enzymes do not. And so if you're a chemist and you want to make tyrosine A, you would probably take a solid phase approach. So in SPSS, solid phase peptide synthesis, you would go and have some resin, and you activate it with an amino acid, and then you would go and take this resin and run it through a series of repetitive reactions to go and build up the peptide on the resin. And so this is a really nice strategy because you're able to go and use automation often. You could buy a solid phase peptide synthesizer. And so this really allows for high throughput generation of different peptides. What's also nice is this idea of a split pool system. And so, for example, in the top case, you don't have to use all your resin and conjugate just a single amino acid onto the end of it. You could split it into two, and then in one case, you could add a tyrosine. In the other case, you could throw on a valine. And so in this case, you're able to generate 6 dipeptides through only 9 reactions, whereas if you didn't use this split pool idea, you'd have to use 12 reactions. And so you can imagine that this gets exponentially more important and allows you to access even more chemical space once you make really long peptides, and as in the case with tyrosine A. And so if you want to generate tyrosine A, you could have your enzymatic and your solid phase approach, and both of them are very valuable. And in the case of the enzymatic approach, you're going to use an NRPS, and it's very good at making tyrosine A and only tyrosine A. You're not going to have a 9 amino acid ring cyclize. You're not going to accidentally iterate and make an 11 amino acid ring. You're going to make tyrosine A. And so this is really good in terms of cyclizing and making this 10-membered ring. But if you take a solid phase approach, it's very nice because you could go and build up many analogs on the resin and not have to really worry about messing up the system. In the case of an enzymatic approach, if you try to use an SNAC or SFP to jump into the middle of the assembly line, then you'd have to worry about the efficacy of the condensation reaction. While if you take a solid phase approach, it doesn't really matter what amino acid you're building up. The reaction is going to be the same either way. But with the solid phase approach, at the time, the cyclization reaction wasn't always generalizable. And so Chris asked the question, these are 2 great approaches to making tyrosine A. How can you merge chemistry and biology and take advantage of both of these systems' strengths? And so what Chris, Rahul, and Mike Burkhart did is they went and took some resin and they appended a chemical linker to it. And what you notice is they took inspiration from the phosphopantethine arm, and they went and built up an arm that looks like a phosphopantethine arm. They then went and added this to a solid phase system, and then they were able to fortunately generate tyrosine A. And then they were able to use the solid phase system to go and generate many analogs very quickly. And so they asked, how does changing residues 1 and residue 4 affect the antibiotic efficacy of tyrosine A? And so looking at the bottom right corner, the authentic tyrosine A standard, you can see that if the y-axis is an indication of its therapeutic utility, you have many improved analogs. And they're able to generate over 300 analogs very quickly. And this was a great example of taking a chem-bio approach to not only making better drugs for human health, but also able to gain insight into TE enzymology and understand how different residues play a role in macrocyclization or hydrolysis. And so I'll pass it off to Rahul one more time.
Thanks, Alex. So we just want to end on a note of, our intent here was to kind of build up and understand that simplicity, again, that exists in the NRPS assembly lines, and then to recognize how chemistry can be brought to bear on these systems in order to find ways, surrogate ways to simplify the enzymology that's here. There were a lot of things in these systems that I think were important to Chris, and questions that are still open ones that he left us with more work to do on these fronts. The idea about individual transformations within the assembly line, revealing new kinds of chemistry, new classes of enzymes, new transformations inside of the molecules that are there in nature in this natural product biosynthesis. I think there were a few things that were more exciting to him than the idea of discovering new chemistry that nature had to offer. And the idea that there is more yet to be discovered, especially in an era when assembly lines can be now easily identified given genomic information, leaves us with many open questions about what else is out there in nature to be discovered. The idea that then, if we want to take that logic, and if we understand how a system works, that we should be able to build new molecules is, I think, still the major and big open question that inspired Chris, inspired many others in this field. How is it that we can take our understanding of a system, do we understand it well enough that we can build molecules from scratch, or that we can take a molecule that exists and tailor it in just the right way in order to access activities that might not exist already in the natural products, or might not exist at all in, out of the realm of natural products. These are the kind of open questions that I think that this general approach of thinking about what's happening inside of enzymes and about merging chemistry together with the enzymology, Chris left us with. So again, I hope we've achieved some of our goal in here, and I think Alex and I want to thank organizers for giving us the opportunity to come up with this presentation and to discuss natural product biosynthesis from the lens of Chris Walsh. I think I want to thank Alex for all the hard work that went into this, because for me and us in preparing, it was also a good journey of going back through memories and reconstructing pathways that are here. And the idea that someone who went through the lab and experienced it can impact those who may not have had the time, who may not have been in the lab but knew of Chris, is I think one of the added purposes of this format and what can be achieved here. So this picture here is from around 2014 when Chris was awarded the Franklin Medal in Philadelphia. That's why he's got that ribbon on like a show pony. This is the only time that I've worn a tux since my own wedding to go to the celebration for this. It was remarkable to see that sort of that recognition, that awards that came to Chris from the science that he did. All of us getting together here to recognize him was also, I think, is important, because it sort of says the impact that he had on us and on science. But I think Chris, one of the things that's important to take away from this teaching is that in the end, what Chris was most important to him was this idea of us all pursuing science together, of discovering, of finding a group of people who are as curious as you are, who want to learn as much as you do, who are ready to be taught also. And that was something that we got to experience being in the lab, and by extension, I think all of you get to experience here today as well. On this list of thank yous is a thank you to Ralph Lambalot for that video, because for those of you who are in the lab, hearing that story about Chris, I think you could probably picture him in the lab leaned up with his arms folded and telling that story. And the end of that story, he says, Chris sort of just puts his hands out and says, he gave him the information. He says, do with it what you will. And I kind of picture that that's where we are right here, right now. Chris has left us with so much, the imprint of what he did, and he's throwing up his hands and saying to all of us now, do with it what you will. For all of us who were his trainees or weren't, were impacted in this field. And so, thanks again.
Questions for Rahul and Alex?
That's just fantastic. Thank you for teaching us about that. One question that comes to mind is, I know for a lot of this early work, it was difficult to sort of understand how the modules worked together because there was not much structural biology. Could you comment on what's right now the state of the art in having three-dimensional structures of these assemblies?
Yeah, I'll say that there are others who probably can comment on it a lot better than I can, having lived through a time when there wasn't a lot of structural information. But that idea of building up the way the field got built up was module by, I should say, domain by domain understanding. So individual domains were solved, individual T domain, a PCP domain. Then built it up to 2 domains together to understand how they work together, how they can start to communicate together. I think the field has come a good deal further. I'm sure that there's others in the crowd who can say, what they think of as the pinnacle of our structural understanding. My understanding, though, is that there's, to understand really how assembly lines fully talk to each other, that goal that's still there at the end, combination of structure, of mechanism, and and of enzymology still needs to happen to really bring us to that level of understanding.
Yeah, I just add, basically ditto. We're now able to understand individual parts, but I think we're still scratching the surface on how all these parts come together and work together in sync as full modules, and then not even full modules, full peptides, multiple modules on a single protein.
Any other intrepid question? One other hand. Let's do this, and then we'll go for our coffee break.
Thank you for the wonderful talk. It's amazing that the system makes such a diversity of molecules. I wonder how the system decide when to terminate on this on the peptide chain, for example.
So we showed you a couple of different examples of different ways that termination can take place. So as you move through the assembly line, the module structure is going to dictate unloading and the last sort of step in biosynthesis. The times when it becomes really intriguing is when there is oligomerization that's happening. That's where that question really comes to the fore, right? So the example of enterobactin, it required trimerization before you saw cyclization occur. In the example of gramacydin, it was a pentapeptide that gets built up. We didn't walk through that example. Dimerizes to make the cyclic decapeptide. So the logic of what is possible there is in part going to be dictated by the thioesterase domain and its chemistry. And part of it's going to be dictated by chemistry of the molecules themselves. So in the case of enterobactin, is the molecule such that the chemistry is right for that cyclization to take place, for the serine hydroxyl that needs to be there to be in the right place in order to catalyze the last step of cyclization. So you can, in fact, find intermediates that differ from that, the system where you might have an extra repetition or one short and unloading of a dimer that hasn't cyclized. Those intermediates can be found in biosynthetic pathways, and it's the thioesterase specificity and the chemistry of the molecules that likely dictates that the most desirable outcome is what happens most of the time.
That's good. Let's give all of our session 2 speakers a round of applause. And please enjoy the coffee break.
Thanks, Michael.
Yeah, so we'll be getting back here together at 45 past the hour. If you are Walsh alumni, please come to the stage for a photo. If we seem to have quorum for all Walsh alumni. Thank you. This isn't the clicker. There's no laser. You're with Jonathan.
Mic people up beforehand.
Yeah, they do.
So they have like 4 of them.
I believe so, yes.
I hope not.
Maybe they didn't. Maybe people. Oh, I think people are
those
coming.
guys back there
All the way in the back about mics. Yeah.
Thanks.
Yep. Okay. Walsh alumni, calling all Walsh alumni. Yeah, and also if you are alumni of Chris's alumni, so grandchildren also count, great-grandchildren. Okay, if you are alumni, please come on stage for the photo so that you could also get some of that coffee break. Don't be shy. This is also for those who might be in the lab of Walsh alumni. So if you're in Beth's lab, Beth Satley's lab, Michael Fishback, Ellen Yu's lab, the full generations of trainees could be in this photo, of course. Yi, also come on.
Michael, do you want to be here? I think there's room. There's a good spot right here. Can you short person?
I'm gonna be kneeling in the front here. So like the shot. There we go.
Who's cameras? Who's Rebecca? Who's Rebecca? Oh, that one. Carolyn's taking a picture too.
Thank you. That's gonna get sent around the lift, right?
I don't know either. We'll see. At some point. There's a grandchild. There's no way he's a grandchild. Oh yeah.
Thank you, everyone. We have about 10 minutes. Hi, everyone. We have about 2 more minutes before we get started. 2 minutes. So if you need any snacks or beverages or restroom, we have 2 minutes.
Okay. All right. We're ready to begin our third and final session of the teach-in. In this part of our day of learning, we are going to hear talks that focus on drug resistance, particularly antibiotic drug resistance, as well as using principles of enzymology to design inhibitors of enzymes using rational principles, which has been really pivotal in the biopharmaceutical industry. Chris Walsh, was every major biopharma company's favorite consultant. Once he and I were in a car somewhere, driving someplace from some event, and the subject of moving jobs from one institution to another came up. And, he was, of course, one of the wisest and most grounded people that any of us knew. And so I asked him, I said, did you ever find yourself in a situation where you had an opportunity to make a big career move and you turned it down and regretted it? And he said, yes, there was one. And Diana, I don't know, you can probably guess this better than I would have known, but apparently he was offered the head of research at Roche. And he declined that opportunity and decided it was better to have the freedom and flexibility of an academic where you can have, in principle, a much broader influence beyond just one, though rather large, pharmaceutical company. And that was interesting. And so we talked about, why he had declined that opportunity. And, I'll never forget that conversation because a lot of that wisdom stayed with me and I applied it in my own decision making. So now, let me introduce the first two speakers of the third session. Our first is Professor Jerry Wright. We are going to be hearing his talk electronically. He is at McMaster University in Canada. Jerry was a postdoc in the Walsh lab back in the early 1990s, and he worked on understanding resistance mechanisms against antibiotics, most importantly, vancomycin resistance, which of course is one of the worst combinations of words a person can hear when they have a bacterial infection, since vancomycin is kind of one of the last lines of treatment for many infectious diseases. Now, Jerry is a distinguished professor. He is one of the Tier 1 Canada chaired professors, a very prestigious title. His lab works on a number of different problems in antibiotic natural product synthesis and bioactivity. He holds a number of important posts as a consultant, as an administrator, as even an editor for ACS Infectious Diseases. But what I like best about his biography, at least on his website, is Jerry has a so-called Batman job, which I interpret to mean metaphorically the job which is in the wee small hours of the night when nobody's looking. He's the drummer in a band named Null Hypothesis, which is great, which he describes as a pants-dropping, hard-rocking, booty-shaking cover band. I don't know if we're going to see a whole lot of booty shaking in his presentation, but without further ado, let me introduce him, and when he wraps up his presentation, I will then introduce his partner speaker, Theodora Brun. So stay tuned for more on her. Are we ready to roll the video? All right, thank you.
Good afternoon, everybody. I'm Jerry Wright. I'm at the McMaster University here up in Hamilton, Canada. I want to thank everyone for giving me the opportunity to speak at this event. I'm really sorry I could not be there in person because of some family issues that have come up, but I really am grateful for the opportunity to share a little bit of my experience being in Chris's lab as a postdoc from 1990 to the end of 92, where I worked on vancomycin resistance. So I'll just share my screen here. And get started. So, as I said, I worked on vancomycin resistance in Chris's lab in the early days. And the organizers have asked me to try to connect the dots with Chris and antibiotics research that has been ongoing in the lab. So how did Chris actually end up working in the area of antibiotics to begin with? How does someone who is, a chemical biologist or trained as a chemist and a really outstanding mechanistic enzymologist end up writing this seminal book and in 2 editions on antibiotics? What's the pathway to get there? In talking with Chris over the years, I have a pretty good understanding, I think, of how this happened. Probably, like everything, there's a bit of mythology in it, and that's, I'll take full responsibility for that. And if I'm omitting some of the actual history that happened. There might be others in the room who know it better, but this is my understanding of how Chris got into the area of antibiotics. I'm going to try not to have too many of these really cheesy Beatles titles, but I think it starts with what I call the magical mystery tour of the cofactors. So we all know that Chris wrote this unbelievable book on mechanistic enzymology that was my gateway into this field, and it really was, you know, an amazing application of incredible discipline and systematic understanding of how enzymes work. And it starts, I think, with riboflavin as one of the first cofactors that he looks with, and that of course does have a connection to resistance. In this case, it's through the Mur operon and resistance not to an antibiotic but to a toxic atom, a mercury. And somewhat tangential, but I'm sure the thinking of the logic of how resistance is connected to enzymes starts to happen. You know, a little bit later, he's working on another really important cofactor, pyridoxal phosphate. You know, launching into suicide substrates, that the chemistry around these really interesting molecules can be leveraged for the development of molecules that could probe the mechanisms of these enzymes and maybe even act as leads for drugs downstream. And the key enzyme here that starts to link this fundamental work on cofactors to antibiotics is alanine racemase, an enzyme that inverts stereochemistry at the alpha carbon here of this vital amino acid, converting it from L to D-alanine, which is an essential component of the bacterial cell wall, moving us towards targets for antibiotics. So the connection between pyridoxal phosphate, alanine racemase, and antibiotics, I think, starts here. This is a paper from the team at Merck Sharp and Dohme, where they, in 1972, prepared fluorinated alanine derivatives that were shown to have antimicrobial activities and antibacterial activities, and some hints about acting on the cell wall. I know that Chris consulted for Merck for many years, and I think that's probably the entry point for him, thinking about pyridoxal phosphate, suicide substrates, which the beta alanines were, and then moving into this area with the lab. So a critical step in this direction is a collaboration with David Botstein's lab, where in the early days of cloning, 2 alanine racemase from Salmonella were cloned, and the beginning of the enzymology was explored. This was followed up by a series of really outstandingly creative and exciting studies by Steve Faraci and Ken Duncan on mechanism-based inactivators of alanine racemase that really sort of set the stage for moving this fundamental research on cofactors towards thinking about antibiotics and the role of these enzymes playing cell wall. So this is just to remind you a cartoon of the cell wall biosynthetic pathways. This is what's happening inside the cell. Alanine racemase catalyzes this reaction, L-alanine to D-alanine. This is then converted next to D-alanine, the dipeptides, through an enzyme called D-alanine ligase, ATP-dependent enzyme, and then grafted onto the growing cell wall here to form this pentapeptide off of this lactyl group that eventually gets incorporated into a molecule called lipid 2, which holds the subunits, the core subunit for peptidoglycan that's flipped outside the cell and then grafted onto the cell wall as it's growing during cell division. So these enzymes were working together, and the folks in Chris's lab, Betsy Daub and Laura Zawadsky, were the first to explore these dealanine ligases, and then again the connection to enzyme inhibitors, and the team at Merck becomes apparent, where we have these exploration of some of the molecules prepared by them, these phosphonates and phosphonates that were kicking around the lab even when I was there, as a way forward. This enzyme over here becomes really interesting as well. This is MUR-A, which puts this phosphine, or this enolpyruvyl group, which eventually gets reduced to the lactyl group from phosphoenolpyruvate, also a target for antibiotics, this time fosfomycin. This is me as a PhD student. This is where I enter the scene. I was a grad student in John Honick's lab, who was also a postdoc in Chris's lab in the early 80s. And he encouraged me to apply to Chris. I had met Chris at an ACS meeting in Toronto a few years earlier, sent him a letter, like an actual paper, and he returned the letter, invited me down to interview, and we talking about projects. And I was really excited about this one, which was on the table, because Chris was at the time moving into the cell wall, and like anything in the Walsh lab, it's kind of once Chris latches his intellect and ideas on a pathway, on an area, I mean, there's no stopping him. And so the cell wall was the next thing. This is what the enzyme is supposed to do. You have this tetrahedral intermediate. I didn't do any of this work, by the way. Eventually, to get this cell wall precursor, I had written a fellowship on this and designed some synthesis to make a series of fluorinated molecules. And I was excited about moving down to Boston in the winter of 1990, when I was finishing up work on the corrections to my thesis in December, and I get a phone call in the lab. This is a phone, that's what they used to look like for the youngsters in the audience. And by the way, Chris always denies this. And those of us who know him, it's like Chris will never actually call somebody like this, but he did. That's the actual thing that happened. And he said, listen, Jerry, I know you wrote about this fellowship about phosphoenolpyruvate, but we just stumbled into this new program working on this antibiotic called vancomycin. And would you consider working on vancomycin instead? And of course, I was wet behind the ears, newly minted PhD student or graduate. I'm not going to say no to Chris. I said, of course, absolutely. And I immediately ran to the library to figure out what vancomycin was. And all of this came from an absolutely remarkable discovery by this guy here, Patrice Crevalin at the Pasteur Institute, who was one of the pioneers in the area of using molecular biology to study antibiotic resistance. And what he had realized is that there was a new challenge emerging in the clinics, which was resistance to vancomycin and enterococci. So we now know this as VRE, which is now probably in every hospital on the planet. And it was quite a mystery at the time. No one knew how this could occur. Lots of people had thought that vancomycin, you could not get resistance to vancomycin, but lo and behold, here it was. And then Patrice's team identified all of the genes and this transposon that were involved in it. One of them, he and his team realized when they cloned it, was a analog or ortholog of alanine, dealanine ligase. So that's where that connection comes. And I think, as memory serves me right, this is Laura Zawadsky reaching out to the Crevalin lab. And so that started a collaboration between Team Crevalin and Team Walsh that resulted in a couple of pretty important papers, I think. The first one was led by Tim Bug, who was a postdoc in the lab when I got there, to show that indeed is a dealanine ligase, but had a weird substrate specificity. And then at my joining the lab, soon after we were able to understand that it's actually making a depsipeptide, dealanine D-lactate. And I was able to synthesize some of these depsipeptides and titrate the cell wall precursors with the antibiotic and show that there's a huge difference, 1,000-fold, between the binding of them. And you're going to hear more about this with Theo's talk. So again, in the typical Walsh lab fashion, a search and destroy opportunity occurs, and we went to work on all of these enzymes. I was luckily enough to be involved in exploration of most of them, but you don't do anything by yourself. And so I worked with Ted Holman on the 2-component regulatory system, and this was followed up by outstanding work by Stu Fisher. Tim got me started in this, and then in the ligases, and then Ilson Park, and then later on Vicky Healy did a lot of great work. And then Zen Wu and Yvonne Lassard and Dewey McCafferty followed up on some of the earlier work we did on Van X. None of this, of course, is done in a vacuum, and then the team had outstanding collaborators from the outside, Jim Knox, who solved the structure of the alanine-dealanine ligase, Barry Warner, who helped understand the regulatory systems, and Dan Kahn, who was involved in a number of downstream projects trying to understand the induction of the system as well as the importance of glycosylation of some of these proteins. I think it's important to realize that when I joined the lab, it wasn't, it wasn't studded with tons of people working on antibiotics. In fact, I was the only person until Ted showed up that was working on antibiotics. In fact, people were working on all sorts of things. Lots of people working on cyclophilin. They were working on trypanothione biosynthesis, photolyase. The mercury operon was still working incredibly well. Ethan Kouliopoulos was working on this crazy enzyme called vitamin K-dependent carboxylase from enormous livers that he was hydrolyzing in the cold room. There was a little bit of biosynthetic work that was happening on enterobactin biosynthesis, but the lion's share of what was going on was really a lot of work on kinases and phosphatases. About 2 to 3 months after I joined the lab, Chris held a meeting and said, I got some news, and everyone said, oh my God, we're moving to Stanford. And he said, no, what I've done is I've decided to take on the presidency of the Dana-Farber Cancer Institute in addition to being chair of the department. And so that meant that we were going to do a lot more work on cancer. In fact, we had a lot of brew meetings where people from the Farber came and told us a lot about cancer. It's a fascinating time. So at the end of all of this is my time to go back to Canada. Chris took me to the Hilton in Back Bay for the goodbye lunch. I don't know if he still did that afterwards, but it was one of the traditions that he had. He would take people leaving the lab out to lunch for a one-on-one. It was great. And he asked me what I was thinking of working on as a newly minted assistant professor. And I said, well, I'm really interested in antibiotics and resistance and how enzymes fit in. And because there wasn't a lot of work being done on that area. And he said, oh, that's a great idea. And I said, I'm also really interested in this molecule vancomycin. It's such a weirdo. How is it put together and stuff? So I think I'm going to start trying to understand how vancomycin is biosynthesized without any understanding at all about what that meant, by the way. It just seemed like something I could do. I figured I could grow bacteria, and I was pretty decent chemist. I could probably figure this out. It turned out to be a lot harder. But of course, and at the time, and this is true, Chris said, that's great. We're not interested in antibiotics after the vancomycin resistance story gets all ironed out. I'm going to focus on cancer. That's where things are moving. And of course, that's not what happened. This is just some of the amazing molecules that Chris, then in the next phase of his career, basically, again, with the Walsh approach, laid waste to the field, essentially, and worked with many of you in the audience to try to understand how antibiotics, natural products, and applying mechanistic enzymology to these things and chemical logic can be used to understand what's going on. I don't have hardly any pictures. There wasn't phones at the time of this. This is a picture of me and my colleague here at McMaster, Eric Brown, who was also a Walsh postdoc. He actually did do the work on Murray. And we were at, this is 2005, we were at the Canadian Bacterial Diseases Network meeting, and this is Chris. He gave a keynote there. I was delighted to host him, and really, it's hard to imagine a scientific world without Chris Walsh. But I'm glad that we're here, that we've got the memories and the training that he provided us. And I'll stop there. And thank you again for the opportunity, and turn it over to Theo.
And it's my great pleasure to introduce Theo as our next speaker. And by the way, if you have questions related to Jerry's presentation, Theo would be delighted to answer them for you, as she's just learning in this moment. But Theo is an MD, PhD student at Stanford. She's an aspiring physician scientist. She is a member of our Seraphan Chem-H, Chemistry Biology Interface predoctoral program, so she's a CBI student. In addition, she holds one of the prestigious Knight Hennessy Scholars Fellowships, and she's doing her PhD work in the lab of Peter Kim. She has many claims to fame as a graduate student. She's very involved in vaccinology and new vaccine development. But today, in line with the theme of our teach-in, she is going to talk about designing enzyme inhibitors using mechanistic considerations relevant to antibiotic and drug-resistant strain treatment. With that, I'll turn over the podium.
Perfect. Thank you so much. It's an honor to be here, and it's been so wonderful to get to know Jerry and learn so much about Chris Walsh's legacy and the impact of his work. So I'll jump right in to talk about vancomycin. So vancomycin was isolated in 1957 in a jungle in Borneo by an organic chemist working for Eli Lilly. And they immediately noticed that it had amazing bactericidal activity. So it's active against gram-positive cocci and bacilli, and also importantly against MRSA, methicillin-resistant Staphylococcus aureus. Its name, vancomycin, comes because of its amazing bactericidal activity. It's able to vanquish its enemies. And so it's currently still one of the oldest antibiotics in use. It's been in clinical use for close to 70 years. It's on the WHO list of essential medicines. So it's really an important antibiotic currently in healthcare today. So what it does is it actually targets the cell wall. So gram-positive bacteria have a thick peptidoglycan cell wall. And the way that they make these thick layers is actually by cross-linking peptides that are on the peptidoglycan chains. And this forms a thick cell wall, which actually gives gram-positive bacteria their name, because when we use gram stain, the peptidoglycan wall retains that violet dye and gives it that color. And what vancomycin actually does is it binds to the terminal portion of the peptides, and it prevents cross-linking between the growing peptidoglycan layers. And so it inhibits cell wall synthesis, which renders the cell very weak to osmotic pressure, and you get leakage of intracellular components and eventually death. So vancomycin is really effective at binding to this portion and stopping that synthesis from occurring. Now, after about 30 years of clinical use, around 1988, we started seeing the emergence of vancomycin-resistant strains, in particular in Enterococcus species. And so, as Jerry kind of mentioned, something that right around the time when he entered the lab, there'd been some sequencing done which had revealed these two important genes, VAN-A and VAN-H, which were clearly critical for resistance, but it wasn't entirely known how. So what we did know at the time, or what Jerry knew at the time, and when this paper that I'm going to talk about really was focused on, is it was known that VAN-A could synthesize dipeptides diala in the format of diala-DX.
But it wasn't clear what Van-H did. And it also wasn't clear what the link was between the two. So first, what they noticed is that Van-H actually had a lot of similarity to lactate dehydrogenases. So this might bring us back to biochemistry 101, but lactate dehydrogenases are ubiquitous in cells. They're a critical part of metabolism, in particular glycolysis and gluconeogenesis. And similar to the lactate dehydrogenases found in other bacteria, Van-H had that motif at its N-terminus. And so that gave them a clue as to what Van-H might be doing. And when they then went on to look at some kinetics, they saw that similar to other LDHs, Van-H was able to reduce pyruvate and 2-ketobutyrate, but also actually had a wider scope of substrates that it was able to act upon. So it's clear that Van-H was likely evolutionarily related to other lactate dehydrogenases, but actually had this expanded scope of substrate specificity. So bringing us back to this paradigm, we now knew that Van-H was able to synthesize these dehydroxy acids, but the link between the two genes was still not clear. Now, it turned out that Van-H was actually able to use the products that Van-H synthesizes and form these dipeptidases. So here we can see the products using these alpha-hydroxy acids. Van-A is actually able to form a series of dipeptides. And the important thing is because it's using alpha-hydroxy acids and not amino acids, this important link is actually now an oxygen atom and not a nitrogen. So instead of that amide bond, you have this ester bond. And that will become very important in a moment. So to then test not just in vitro, but actually using bacterial extracts, they use a very critical reagent. So we heard a little bit earlier about the importance of radioisotopes, and that's what they did here. So using isotopically labeled precursors and then combining with Van-H, what you're looking at is a chromatogram. And you can see there the different products formed in that dipeptide format. And then they took crude bacterial extract from an Enterococcus line BM4147, which contains the plasmid expressing Van-A and Van-H. And you can see here that in that crude extract, they were actually able to see the formation of these alternate Diala-DX products. In comparison to taking a bacterial crude extract from a line that doesn't have Van-A or Van-H, you don't see those forming. All you see is the formation of Diala-Diala, which is the normal product that's expressed. And interestingly, if you look at the line corresponding to the formation of Diala-Diala, you can see that that's actually suppressed in the lines in the vancomycin-resistant bacterial extract, suggesting that there's actually a way that it suppresses the formation of Diala-Diala while making these new dipeptidases. So now when we look at this tree, we see that there's a clear connection between the two because the product that Van-H makes is a substrate for Van-A. And together, they form a new type of peptidoglycan. So vancomycin, unfortunately, very, very good at binding to Diala-Diala. We're in the micromolar range here, but actually does not have appreciable affinity for these other dipeptides. And the reason for that comes down to the substitution of that amide bond for an ester bond, and also the presence of some of these bulkier side chains. The way that vancomycin binds to its substrate on the peptidoglycan wall is actually through the interaction of 5 hydrogen bonds shown in dotted lines. And shown another way here, we can see in this construction that critical hydrogen bond with the nitrogen. And then when you have the loss of that and the replacement of an oxygen, you actually lose that hydrogen bond. And this is the reason that vancomycin is unable to bind these new peptidoglycan chemical structures. So in conclusion, we now know that we know what these mobile elements do.
They can be transferred among bacteria, which is how resistance spreads. And through the action of VAN-H, the alpha-keto reductase, and VAN-A, which uses the products from VAN-H, they form a novel peptidoglycan structure that vancomycin is unable to bind. And so I wanted to end with this quote from the very last paragraph of the paper, which basically says, an understanding of the molecular basis for glycopeptide resistance could then lead to the development of anti-resistance agents and perhaps the development of novel irresistible glycopeptides capable of binding these new peptidyl diala-DH-bute products. Unfortunately, even though this paper was published in 1991, today we don't actually have any such products. This is a graphic from a paper published just last year in 2022. And we can see that some of these major resistant bacteria are still circulating today. In fact, this is a picture of me on my first day going back into the clinic, into the hospital after finishing my PhD, going into the lion's den, the hospital, the heart of vancomycin-resistant enterococci. And so I hope that today, and listening to Chris Walsh's work and his legacy, some of us may be inspired to look for some of these solutions and some of these new antibiotics. Thank you.
Hi, thanks. I was just wondering if the bacteria that are resistant by this mechanism, do they pay a cost? Is it not as good a cell wall? Do they?
Not that I know of, and I'm curious if our infectious disease specialist has insight into that. But I think it's actually an effective mechanism, and the altered enzymes actually have very high affinity for these substrates, and so not that I know of.
Thank you for the great presentation. Can you tell us about the origin of the VAN-A and the VAN-H genes? Are they from the Enterococcus genome, or are they from some other species?
I believe that they were first sequenced in Enterococcus strains. They're found on plasmids that can be transferred between them. And so I believe that that's where they were first discovered. The scary thing, of course, is that there are cases, if you have a patient with VRE and MRSA, MRSA can actually acquire that plasmid and become vancomycin resistant as well. So they've certainly spread. But I believe that was the first occurrence.
Oh, I have a question. So you said, there was the proposal by Chris Walsh and his coworkers back in 1991 that if we could figure out how to make a vancomycin analog that recognizes that types of peptide, maybe we'd be back in business. We're so good, aren't we, at designing molecules that interact with specific substrates and ligands? And what have we tried that has failed?
Well, since we don't publish negative data, I think it might be harder to know. What I do know is a lot of work and a lot of vancomycin derivatives have been focused on extending half-life, making them more bioavailable, enhancing their affinity. So that's kind of as I was tracking down what's kind of happened since this paper was published, that's what I was able to see. And I'm not sure why we don't have a better vancomycin for this altered substrate. One of the issues with antibiotic research in general is, from a pharma point of view, the incentives are quite low. If we do make a new antibiotic now, it's put on a shelf and locked away, and we're saving it as our last line agents. So there's no immediate payout compared to, say, a cancer drug. That's kind of from an incentive point of view, but from a science point of view, I'm not sure, and I wish I knew more.
I think your question highlights another dimension of Chris, because the answer to your question is, when Chris started to recognize that the problem you think should be straightforward was not really as straightforward, as one might, he also recognized that there might be alternative solutions. And one in particular that he recognized quite early was the possibility of using the glycosyl groups on these antibiotics to modify the regulation of these pathways of resistance in these resistant organisms. And that's when he came across your work, that's when he came across Dan Kahn's work, that's when he came across the work that led him to persuade Dan to basically change his career. And so that's a very common pattern in the Walsh legacy.
Yes, Jim. Take this one.
Thank you, Carolyn. That was a wonderful talk. I just wonder, how are the bugs making vancomycin? How are they resistant? Do they not have these structures in them? It's an extracellular molecule, so maybe they would be vulnerable.
Well, so they are vulnerable if their cell wall is built with diala-diala. And vancomycin as a drug is an IV drug that we use in the hospital. So once it's there, it will do its job. But I think because of the ubiquity of, initially people were very surprised that vancomycin resistance could even emerge because of the ubiquity of alanine and its importance. And it was very hard for people to envision how anything else could be used in the place of alanine. So I think now it seems so obvious, like, oh, of course, just change your peptidoglycan composition. But I think this paper is one of the seminal papers because it really was a paradigm shift in the way people thought about what bacteria could do. And it just goes to show that given enough time, resistance will emerge. And even before resistance emerged, there was vancomycin creep, which was the need to use higher and higher and higher doses of vancomycin. So I guess it's constantly occurring, this evolution in the background, low level, even without getting to the point where you have these genes that are being transferred.
Why do you think there's so little diversity in the diala-diala part of the peptidoglycan cell wall? Like, it seems that, as you say, there's not a huge fitness cost to changing it. So why so little diversity?
Yeah, I think that's a great question. I think based on the chemistry, it is a perfect substrate. And I think mechanistically, it's actually quite interesting that we're able to get this altered substrate specificity even in the presence of alanine still there. So I think even though it seems obvious, it's not actually that trivial from a chemistry point of view, which we didn't necessarily. So that would be some of my thoughts upon the matter, and I would welcome other expert suggestions.
I mean, one approach that has been tried, and this, which was founded by Matai Mammon, who many of us know, and also John Griffin, who was a Stanford chemistry faculty member and might have been a Walsh alum. I'm not even sure, is that true? He was a Walsh postdoc. And their idea was, if vancomycin binds more weakly to this altered diala-D-lac sequence, what if you dimerize vancomycin and exploit multivalency to compensate for that? And in the end, my understanding of that story is it was just too difficult to manufacture dimers of something as complicated as vancomycin and make a drug out of it. This was before click chemistry. But enough about me, I guess. Yeah, all right. Thank you so much. Please join me in thanking Theo and Jerry, both. All right. Okay, so which brings us to the final session. Very exciting for me to introduce our last reunion kind of situation. Woohoo, yeah. So, so first we have Michael Marletta, who is a distinguished professor at Berkeley. His lab has made a number of seminal contributions in enzymology and physiology around nitric oxide synthase enzymology, more recently enzymology related to breakdown of cellulose, which is an important process for renewable energy sources. He just told me, was postdoc number 4, I think, in the Walsh lab. So he was there, you know, at the very beginning, and then went on to have an illustrious career where he has been one of my role models and mentors for many years now. He's going to kick off the session and then very quickly pass the podium to Jonathan Mazza. Jonathan is now a postdoctoral fellow. He holds a Hannah Gray Fellowship in the lab of Jim Wells at UCSF, but he did his PhD at UC Berkeley as a member of the chemical biology graduate program there in the lab of Matt Francis. So let me first invite Michael and then also Jonathan to come up to the podium, and then you can take it from there.
Thank you, Carolyn. So yes, you know, I think it was Chris who said to me once when I was thinking about breaking the rules, that he said, you know, treat it like a stoplight in Boston. It's just a suggestion. So we're going to mix it up a little bit. It's the last one of the day, so we probably need a little bit of shaking up. I'm going to have a few comments here to set the stage for Jonathan, and then I'm going to break the rules, I suppose, and come back in the end and make a few comments, a little personal reflection on Chris. It's the last talk of the day. You know, I got the mic. That's the way it is. Chris did call me once, and then when he called me, some of you might remember the phones used to have like a little round thing on the front of them and had holes in it, and you could put your finger in the hole and move it in a clockwise position until it came to a stop, and that's how you used to make phone calls. Right, so I guess it's me who can do this. There we go. So it's sort of fitting here at the end, I think. We've had a focus on Chris's entire career. And we're going to sort of make a circle and come back to the beginning. So there were the 3 phases of his career, MIT, Harvard, and then Stanford. I should probably make a comment here about, he didn't just write books at Stanford. In fact, I was in the lab the day this one got published, the one that served as a gateway for many of us, as Jerry Wright said. Chris, you know, knows that he got a phone call. They had a package on the loading dock. And he went down to the loading dock, and he picked up this big box full of these books, first box that was shipped out, put it on top of his head, and walked through the lab, delivering copies to those of us that had taken the time to read various chapters. So it was a really exciting time to be in the lab. I did my best at the arc of his career to pull out the highlights, and you have seen many of those. We're going to return back to the early days, in part because you could simply ask the question, why study mechanism? I mean, there's the beautiful chemistry you can uncover. We've seen that all day. But you can do something with mechanism. And we heard a little bit in the very beginning about mechanism-based inhibitors, and that's what Jonathan is going to talk about. So we're back to alanine racemase, and that's what Jonathan's going to talk to you about. Last time I checked, there are 20 naturally occurring amino acids, so we've been focused a lot on alanine this afternoon, but there are others, as you know. But we're going to come back and focus on that simple molecule, and the stage has really been set for how important it is, at least in prokaryotic growth. And we'll hear about how you can use mechanism to develop a very selective inhibitor. So Jonathan, where are you? It's all yours.
Yes, hello. So I'm Jonathan, and I'm really excited to be here, and I guess just tell a little vignette from Chris's career on these mechanism-based inhibitors of alanine racemase. So as Theo so kindly introduced for us, the bacterial cell wall is a really important target for the design of new antibiotics. And that's because it has this critical peptidoglycan layer that doesn't exist in human physiology. So if we can find ways to target and inhibit that peptidoglycan layer, we can perhaps design new compounds that can kill microbes. And from a chemical perspective, peptidoglycan is quite interesting. It's comprised of these repeating sugar peptide units, and importantly, they involve this covalent cross-link that I'm showing right here that is really critical for providing its structure and integrity. And now, one of the key components of peptidoglycan is this D-ALA motif, which signals to the enzymes that cross-link peptidoglycan to perform the cross-linking reaction. And so the bacteria's ability to access D-alanine is really crucial to its survival. And now, life is chiral, and most amino acids, I think as Michael kind of mentioned, exist in the L isomer. And so bacteria are able to synthesize L-alanine using their biosynthetic machinery. But they don't really have a route to access D-alanine. In order to do that, they're going to need to find a way to convert L-alanine into D-alanine. And the enzymes that bacteria have evolved to do this are called alanine racemases, and I'm showing a structure of the E. coli one here. And the critical transformation that they mediate is taking L-alanine and converting that stereocenter at the alpha carbon there into the corresponding D isomer over there. Now, how is the enzyme going to do this? It can't just grab the amino acid, and twist about that bond. Instead, it needs to break a bond and reform a bond at the opposite face in order to convert between these two isomers. And the critical bond it's going to go after is that hydrogen at the alpha carbon position. And in order to do this, it's going to use a very critical cofactor that we've already heard a little bit about today called pyridoxal phosphate, or PLP. And the mechanism of how this enzyme works is it starts out like all PLP enzymes, covalently tethered to a critical lysine residue to PLP. The primary amine of L-alanine is then going to come in and swap out, and you're going to get now this Schiff base intermediate where L-alanine is conjugated to PLP. And I want to highlight this very critical positively charged pyridinium group in the PLP cofactor that's going to allow the enzyme to accommodate the electrons in that CH bond as it's performing that bond-breaking reaction. And that's what you're going to see here. A basic residue in the enzyme is going to come in, abstract that alpha proton, and those electrons are going to cascade down into the PLP core. You're then going to access this planar quinoid intermediate, which is now set up to isomerize that stereocenter and alanine. And so those electrons are going to rebound back up and ultimately grab a proton from the opposite face from which it started, giving you D-alanine. And of course, this is an enzyme, so lysine comes back in and you regenerate the system and liberating D-alanine. And so this enzyme is very crucial for the bacteria's ability to access this very critical amino acid for its peptidoglycan synthesis. And at the time of this publication, it had been observed that these beta-haloalanines, some of which the work was being done at Merck, had these antimicrobial activities. And at this point, it had kind of been deduced that alanine racemase must have something to do with it, but it was really unclear what the mechanism of inhibition for alanine racemase was for these compounds. And so Chris, with his really great mechanistic detective work, took this up with Elizabeth Wong, and they kind of set out to piece out the puzzle of how these beta-haloalanines were inhibiting this enzyme. One of the first clues they had was looking at how these molecules inhibited the enzyme over time. And so what I'm showing over here is a graph of enzyme inhibition over time. And what they did was treated the enzyme with differing concentrations of these molecules, and then at different time points, take out aliquots of the enzyme and probe how much activity remained. And what you'll notice is this very unique or interesting phenomenon where the inhibition is not immediate. You see this time-dependent decrease in enzyme activity. And so this told him that something was happening mechanistically. The enzyme was still able to chug along somewhat, and that was a really unusual feature. Another clue came when they looked at the PLP absorbance spectra of this enzyme. And so one of the unique features of PLP tethered to an enzyme is it has this really nice absorbance peak at 420 nanometers. And that's what you're seeing in this UV vis trace here in the solid line is the absorbance of the untreated enzyme at 420 nanometers. But when they treated the enzyme with fluoroalanine, they saw a complete obliteration of that 420 nanometer signal. And this told them that something was occurring with that PLP cofactor in terms of how this small molecule might be inhibiting the enzyme. And one final clue came in some previous work the lab had done. And so, as I mentioned, the canonical activity of alanine racemase is to take the L isomer of alanine to its corresponding D isomer. But the Walsh lab had previously observed that treating this enzyme with these beta-chloroalanine derivatives like L-chloroalanine or D-chloroalanine in either case led to the formation of pyruvate and ammonium chloride without any racemase activity. And so this told them that the enzyme, when treated with these beta-chloroalanines, had this alternative pathway. And this same pyruvate formation was observed if you treated the enzyme with fluoroalanine. So what you're seeing here is the formation of pyruvate with respect to time at various concentrations. And what you'll notice is that similar to that time-dependent inactivation I showed a couple slides ago, you see the gradual increase in pyruvate over time, and then eventually the enzyme dies and stops. And so this kind of told them that whatever was happening with these small molecules, the enzyme was chugging along, forming pyruvate before it ultimately inactivated. So Elizabeth and Chris pieced together these key observations. They knew that whether or not you worked with the chloroalanine derivative or the fluoroalanine derivative, the L isomer or the D isomer, in all cases you accessed this planar pyruvate molecule. In all cases, irreversible inactivation was observed, so you couldn't just take the enzyme, treat it with these molecules, and then dialyze to restore enzyme activity or add in fresh PLP and restore enzyme activity. In all cases, the enzyme was irreversibly inactivated. And in all cases, PLP absorbance was lost. And so they pieced together these pieces and came up with the idea that there must be some kind of common intermediate in the mechanism of this enzyme that was ultimately leading to its inactivation. And so they proposed this mechanism here where you have your beta-haloalanine, whether it's the D or L isomer, and they propose that in either case it must be still accessing some kind of symmetric planar intermediate because they saw inactivation in either case. And so they postulated that it likely was still accessing that key quinoid intermediate that I showed a couple of slides ago. And now if X in this case was a hydrogen, you would just move forward and isomerize. But if X was a fluorine or chloride, they instead postulated that the electrons could rebound in this alternative pathway, which ultimately lead to expulsion of that halide. And this led to this very critical N-aminoaldamine intermediate shown here, which could ultimately go down two alternative paths. In one route, you could imagine hydrolyzing that bond near the top there and ultimately accessing that pyruvate molecule that they saw whenever they treated the enzyme with these molecules. And in this case, the molecules would be acting as competitive inhibitors, just occupying the enzyme's time instead of allowing it to turn over alanine. But in an alternative route, you could also envision, if you look up at the top, you have a really juicy Michael-type acceptor electrophile, and you could also envision a nucleophile in the enzyme advantageously adding into that electrophile. And ultimately, through a series of steps, that would lead to an alkylated form of the enzyme that would likely perturb the enzyme's active site geometry and prevent it from binding and turning over D-alanine. And in this case, the molecules would be acting as irreversible inhibitors and ultimately killing the enzyme. And so by taking together these different puzzle pieces, Chris was able to piece out this mechanism for how these molecules might be inhibiting this enzyme. And they're ultimately able to extend this work to some naturally occurring inhibitors of alanine racemase like O-carbamoyl D-serine as well as D-cycloserine. Now these were molecules that were both isolated from Streptomyces in the 50s, and they were being explored by companies like Merck and Lilly for their antimicrobial activity. And importantly, I will say D-cycloserine continues to be used today in the treatment of multi-drug resistant tuberculosis, specifically because of its alanine racemase inhibitory effects. But at the time of this publication, it was thought that these molecules were acting as competitive inhibitors of alanine racemase, and it was Chris's ability to tease out their activity, that he was able to ultimately show that these are actually irreversible covalent inhibitors of alanine racemase. And now there was one final piece of evidence that was interesting to them, and that was that no matter what compound they used, the D isomer always was the better inhibitor over the L isomer. And this made them ask the question of why was there this preference for D isomers? And of course, at the time, they didn't have a crystal structure of alanine racemase, but Chris, using his organic logic, was able to postulate that there must be something unique about the active site in terms of how those atoms are arranged that is leading to some kind of asymmetry in the active site that is allowing, that is causing this preference for the D isomer over the L isomer. And of course, now we do have a crystal structure of alanine racemase, and what you can so nicely see is that there actually is an asymmetry in the active site in terms of the positioning of these very critical tyrosine and lysine residues. And it's just, I think, a testament to Chris's organic logic and brilliance that he was able to kind of see this without actually seeing this. And so the work Chris and Elizabeth did about like 40 years ago really enabled the understanding of the mechanism behind how these molecules are inhibiting alanine racemase by intercepting the mechanistic activity of these enzymes. And this work still has profound impacts today. So it's as recent as 2020 where people were actually looking at the mechanism of inhibition of D-cycloserine for M. tuberculosis alanine racemase. And while Chris had shown that this is a covalent irreversible inhibitor for alanine racemase in E. coli, it's now actually been appreciated that it's a covalent reversible inhibitor for Mycobacterium tuberculosis alanine racemase. Yeah, and so just kind of highlights how Chris's mechanistic organic logic and interpreting how these small molecules intercept the mechanisms of these enzymes has played such an important role in the design of these types of antibiotics. And with that, I'll turn it back over to Michael. Oh, I guess, well, question. You want me to do questions first? I guess I can take some questions first.
You can.
So the enzyme, does it racemize L-alanine, or does it convert it to D-alanine? Like, because from the name, I assume it's making a 50-50 mixture, taking something that's enantiopure and making it into the racemate. But I was just curious.
Yeah, I mean, I think it's ultimately driven by thermodynamics at the end of the day, right? Because it can act in either the L-alanine to D-alanine position or the D-alanine to L-alanine position. And so I think it's ultimately driven by thermodynamics in one direction, if that makes sense. Did I fully answer your question? No. So I guess it's a racemase, ultimately.
I'm curious if, as you were reading the papers, if you got a clue to a mystery that I don't have an answer to. Around the time when Chris was getting interested in this as an exemplar PLP enzyme, there was obviously the other major class of PLP enzymes that was starting to become interesting, the GABA-associated enzymes. I mean, and there was a link, as best as I understand, that cycloserine actually works on those enzymes by then. Is there anything that tells you why Chris didn't think those enzymes were interesting enough for him to take an interest in it?
I think in my conversations with Beth, I think one of the things she highlighted was that Chris always seemed to enjoy the puzzle-solving aspect of dissecting these mechanisms, but always enjoyed if it could connect very clearly to a problem that might impact human health. So, I mean, I guess, I don't know, I mean, these alanine racemases where I think, well, I know one thing, I think this is one of the first projects where he, I think, spoke with Merck and talked with Merck about collaboration. And so I think Merck actually probably popped this idea into his head about, I don't know, the importance of these enzymes, both from an interesting organic mechanistic perspective, but also probably just in terms of importance for human health as well. So that's what, I guess that's why. Yeah.
So
why no therapeutic from this beautifully simple way to intersect bacterial growth?
I think I can't remember what, but I think there's a human diamino oxidase that it actually acts on as well and forms fluoropyruvate. And so I don't know if ultimately down the road that fluoropyruvate proved to just be too toxic for it to be therapeutic. I think it was in the kidney. I think it was the kidney diamino oxidase.
So cycloserine, Chayton mentioned it. So is it still clinically used?
Yes.
I thought it was toxic.
Yeah. It's used for MTB treatment, but also it's used as I think for schizophrenia treatment as well. Yeah. Cool.
All right. Join me in thanking John.
Yeah, so I thought if I would take license, I should say that we organizers, because I was one of them, we all agreed that the organizers wouldn't speak. You can ask Beth why I stand here having the opportunity, which I'm happy to have. So as Carolyn mentioned, I was the 4th postdoc, I'm pretty sure that's right, in Chris's lab, 1978 to 1980. I told him I wasn't interested in a long engagement, and I thought 2 years would be just about the right amount of time. As it turns out, it was actually 2 years to the day. I started on January 1st, 1978, and finished and went on to another position in January 1st of 1980. When I came from UCSF back east to interview, I interviewed with Jeremy Knowles. And Jeremy was extremely smart and very charming. And then I interviewed with Bob Abels. And Bob Abels was very smart. He was not charming. Some of you know him. And then I interviewed with Chris. And I was in maybe 5 minutes into the conversation when I knew that this was the lab that I wanted to join. And I was going to be speaking with him for about an hour before I spoke to the group. And I just tried to concentrate on the next 55 minutes to not say anything stupid so that he would offer me a position. And he did. And I expected, in the time I would spend with him, that he would be a mentor to me. And I expected that I would learn a great deal from him. I didn't expect to have a lifelong friend. And that was just a tremendous outcome for me in the lab. When students join my lab now, I tell them, look, there's only 3 things you have to do. You have to do the work, because we're not a computational lab, a theoretical lab. Actually, you got to mix this thing into this thing and ask what happened. You have to be able to talk about it, and you have to be able to write about it. That's only 3 things. You get those 3 things, you get them done next week, come see me. It hasn't happened yet, but you can always hold out hope. And I bring those up because when I think about what I learned, especially from Chris, so the first year I was there, I sat in on the course that used the enzyme mechanisms book, 550. Those of you with the MIT era will know that course. And I sat in on that course. I watched him teach. I watched him give research lectures, and so I learned how to make slides. I learned how to talk about work, and especially, as I said, I learned how to teach. In the second year, he rarely left lectures that he couldn't. He rarely had a trip where he couldn't give the lecture. And he came to me and he said, I have something I don't even remember what it was. It was pretty important. And he said, I'm going to be away. I'm going to miss 3 lectures in 550. And I want you to give them. And I was honored and said yes. And then I went home and tried to fall asleep. And I thought, what have I done? I'm going to give 3 lectures in Chris Walsh's course. So it was a baptism of fire, but I just thought about how he might do it, and it all seemed to work out. Those of you who know me know that I like to tell stories. I'm just going to tell 2. One of them, yeah, but you don't know how long they are, so who knows? one of them is, it really was an urban legend, I guess, in the lab in those days, that Chris was clumsy and that you shouldn't let him touch anything. So Ralph's story about him coming in and leaning against the bench and using his hands, that was okay. But I had, the beginning of my postdoc involved a fair amount of synthetic chemistry. And I had finally had a molecule in hand that I've been after for a while. And so, he came into the lab, and I told him I had it, and he was excited. And I held it up, and it was in a little round bottom flask. And I held it out as I was talking to him. And as I held it out, he reached out for it, and I pulled it away. And then I continued to talk, and I put it back out, and he reached out again, and I pulled it away. And then he said, "You're not going to give that to me, are you?" And I said, "No." And he said, "Why not?" And I said, "Because I was told you'll drop it." And he said, "Who told you that?" And I said, "Everyone." So when he would come into the lab, you could hear him. He had a sort of shuffle with his loafers, and you could hear him coming. And so I would get up if he was coming near my bench, and I'd do my best act of being an offensive lineman and sort of block my whole bench from him touching anything. I mean, the good news is that I'm not much of an offensive lineman, but he wasn't also much of a defensive end, so it seemed to work out okay. I could block the path. That to me is one of the great stories about Chris and his interactions in the lab. But there are many other ones that you could talk about. I think that when I think of things he's told me and advice he's given me over the years, and I've been lucky enough, 57 years, I just did the math while I was sitting down. It's 45 years ago that I was a postdoc in the lab, so a good deal of time for advice. And I mentioned that he was a great friend. Friends, it's relatively easy, as we all know at this stage in our lives, to be a friend when things are going good. It's substantially harder to be a friend when things are not. And we've all had bumps in the road. I had a fairly big one. And while I was trying to sort that out, I had a very long talk with Chris, and there were some similarities to a bump in the road that he had. And that conversation, long as it was, helped me piece together how life was going to go on and what it was going to look like. And it was probably, and I've had so many scientific discussions with him over the years, but that was the most influential time that I'd ever spent with him. And I would see him a fair amount. I was lucky enough when they made the move to Stanford to have lunch with him every once in a while. I'll tell this one last story. We would come to Berkeley sometimes, and I would come to Palo Alto sometimes. And one time, I had this idea of saying, neither of us have quite the amount of travel time that we'd like to have. So why don't we meet halfway? Well, those of you from the Bay Area know that there actually isn't a lot between here and Berkeley. Hayward. So I found an Italian restaurant open for lunch in Hayward. So I got there a little bit early, and I walked in. There was no one in the place. I don't think they did a big lunch business. And anyway, he showed up. He looked around, looked at me, and we both smiled. I said, "Well, are you hungry? Because I think this is the only option." And so then I insisted. I said, "I want to sit here." And he said, "Why are you so insistent you want to sit here?" I said, "Because Sicilians never sit in an Italian restaurant with their back to the door." He was kind enough to say, I said, they won't care about a Walsh. So, the impact that he's had on science should be clear. The stunning depth and the scope of what he did was beautifully talked about today by everyone who spoke. The stunning depth he had on us that we're lucky enough to be in his lab is also obvious. And I hope what we've done today, and I think actually we've been pretty successful at it from my maybe biased vantage point of an organizer, is that we were able to impart to those of you that only know him through his papers of the amazing human being that he was. It's tempting and easy to say that, you know, after Walsh, they threw away the mold. But I actually think it's deeper than that. There really never was a mold, because a mold implies that you might be able to make another, or there might have been one before him that came out of that mold. He stands alone, and it is absolutely clear from everything you've heard today. I personally have benefited from that. In a few words here, I hope I've given you some indication of that. But I think any one of you who knew him could stand here and tell a similar story about how he impacted your scientific life and how he impacted who you are today. So we owe him an amazing debt of gratitude. So I'm thankful to him. I'm thankful to all of you for all that you both listened to today, and especially those that did their best to describe Chris and his legacy. Thanks.
Thank you. Those are absolutely beautiful and befitting words. Before we close, and for that, I'm going to give the podium back to Chetan just for a few remarks. But first, let me ask everyone to join in a round of applause for all of today's amazing speakers, and especially our trainees. We're incredible. You did a wonderful job of channeling Chris Walsh. Your teaching skills are impeccable. I'm so proud. There's so much to look forward to. And with that, let me turn it back to Chetan to close out the event.
So, thank you for acknowledging the most important people, the 6 odd trainees who presented those amazing stories. Thank you again from me as well, and from everybody. There's a few other thanks that I want to communicate before we close the event. The first one is, of course, to Team Seraphin Chemich. Carolyn can take a bow on behalf of the entire team, it's been an I want to particularly thank Michael and Beth for all of the amazing hard work you did and your unwillingness to settle for anything that was less than superb. It turned out every bit as well as you dreamt, and I hope you can sleep well tonight. Perhaps most importantly, I want to thank all of the Walsh alumni who came to this event. This event would not be what it is without your presence over here. Michael said what Chris meant to all of you through his own lens. Speaking as somebody who's not a Walsh alumnus, I want to just remind you that you all are the guardians of a brand. It's not even a legacy anymore. It's a brand, the Walsh brand, and you are all the guardians of that. What is the Walsh brand? It's a brand that is not just the highest that you can get in scholarship. There's an audacious creativity that you saw in many of the presentations. There's a deep pedagogical value, lasting pedagogical value in that brand. But beyond that, there's a grace. There's a certain level of sophistication, grace, that probably can only be captured in one word that Diana often uses: class. Chris was really a classy guy, and everybody who knew him knows that very well. And so you are all the guardians of that brand. I hope you share it amongst yourselves, amongst your trainees, amongst your co-workers, amongst your own children, because even though Michael is convinced that that mold never existed and therefore can never be reproduced, I'd like to think for the future of this planet, there might be a chance that there would be, may not be in our lifetimes, but in some other people's lifetimes, there could be another Chris Walsh someday. So thank you all for coming to this event and for being part of it. There's one last person I'd like to thank before we close out. Here's a trick question. What would you answer about who's the last Walsh trainee? My guess is most of you will probably call Tim Wencewicz as Chris's last trainee. I've heard that many times, and I love Chris. I love Tim dearly, but he's not the person. Chris's last trainee was no one else other than his own grandson, Sean Kurian, who is with us right now. So Sean, there's two things I'd like to say to you in closing out. The first one is, on behalf of about 200 people in this room, thank you. Because most or all of these people live in the Bay Area. And most or all of them benefited from Chris's presence in the Bay Area over the past 10 years. And there's absolutely no way any of us believe that we would have had the privilege of having Chris as a fellow resident of the Bay Area if it weren't for you. So thank you. The other thing I will say to you is, most of us probably don't think of where our paths will cross with our grandparents when we are at your age. In my own case, my grandfather was a math teacher, and I soon realized I wasn't a big fan of math, so I knew I wasn't going to care about what he was passionate about. But you have the unique privilege that it almost doesn't matter what path of life you choose for yourself. You will have an opportunity to cross paths with your grandfather, because your grandfather is in textbooks of chemistry. He's in textbooks of biology. He's in textbooks of medicine. If there are textbooks of business, he's probably even made it in those textbooks. We didn't do adequate job of representing his legacy in industry, but in addition to the huge number of people over here who are Walsh alumni, who have had a huge impact on the biopharma industry, I mean, Chris was, the word genzyme is, at last I counted, all but one letter, enzyme. And Chris was one of the founding scientific creators of that company. And that was only one of literally dozens of important companies that Chris helped. So chances are, even if you end up pursuing a non-technical career, you'll have a chance to run into your grandfather's legacy. I hope it gives you a chance to get to know him again and enjoy it. So good luck. Thank you.
Okay, thank you everyone. Just one last thing, just a real big shout out to Danielle, who did a lot of the heavy lifting for the organizing all the way in the back. Thank you, Danielle. And yeah, so thank you all for coming. Please get home safe. If you hear EDM music, it's because Frost Amphitheater has a sound check. So don't be scared if you hear loud banging on the way to the garage. Thank you, everyone.